GATK workshop. I'm Laura Gothier. I've been at the Broad for 6 years, and right now I supervise the germline variant calling methods. I've worked on haplotype caller and VQSR and a bunch of the other tools that we have. Right now I'm focusing on structural variation, and I'll let Kate and John introduce themselves as well. Great. So feel free to interrupt with questions at any point. We'll also have time at the end of each talk for additional questions. I know a lot of you filled out the survey, but just to see again, show of hands, how many people are focused mostly on germline? Okay. How about cancer research? Most of you? How about people that haven't started a project yet? Okay, great. All right, so, oh, and then one last thing, Kate wanted to make sure that everyone can take stickers during the coffee breaks or lunch breaks, so you can put them wherever you'd like to do that. All right. So just to give you an overview of what we'll be doing for the whole workshop. So the reason that we developed the GATK is to have a more robust software product than just a whole bunch of Perl and Python scripts that PhD students and postdocs have cobbled together and then stopped supporting. So we wanted to focus on speed, scalability, and versatility. And so that means you can run an analysis on a single sample, you can run an analysis on hundreds of thousands of samples, but it should be possible with the software that we have. So we started our focus on variant calling, but in recent years we've expanded into different types of variants, additional tools, and within the methods group at the Broad, we're also doing work in single cell as well, and some imaging and all kinds of different things, which tools will eventually either make their way into the GATK or another software package. We also recently have put a big focus on reproducible best practices workflows. In the past, we had a verbal description of what our best practices were, but no scripts to run, so you run your own to actually put together what the command line should be. But now, in the new world of Whittle and Cromwell, we can actually provide scripts that you can run so that there's no mistake about exactly what arguments you use and what the parameters are and so on and so forth. And GATK4 is in minor release 4.1, and we're proud to say that all the 4 versions are totally open source. You can go online, you can look at all the code, you can actually fork it, you can contribute. It's just free and open to the community. So GATK has both germline and somatic tools for calling most of the types of variants. Haplotype caller is sort of our star, which is for germline SNPs and indels. UTEC2 is arguably the best in the somatic world for calling SNPs and indels. GATK germline CNV is our pretty much finalized CNV caller on the germline side. And then we also have copy number variation and allelic copy number on the somatic side. Our structural variant offerings are still in progress. We should have a single sample structural variation pipeline to share on Terra within a month or two, if that's something that you're interested in. When we released 4.0, we did a whole blog post series, and so these are kind of the overviews of some of the blog posts that are out there. You can find them by going to that link. But we just talk about some of the improvements that we've made in 4.1 over the initial 4.0 release and over GATK 3 in the past. Like John did a post about Funcatator. I did a post about generating large germline SNP and indel call sets like EXAC and Nomad with tens or hundreds of thousands of samples. There's a blog post for our liquid biopsy pipeline, which some of you may find interesting. Our mitochondrial pipeline we'll look at later in the workshop, and some of the other improvements for our Spark tools and the CNV tools on germline and somatic. So now these workflow scripts describing our best practices for all of our pipelines are available in this public GitHub repo. It's under GATK workflows, and there you'll find all the pipelines that we're going to run as part of the hands-on this week. They're also in Terra, where Terra is our web-based platform for interacting with data in the cloud. And so nowadays, given that there's so much genomic data and it's so large, it doesn't make sense for individual institutions to host multiple copies. So the goal is to have one centralized copy in the cloud that everyone can access. And then rather than downloading the data and bringing that to the methods and the tools, we want to take the methods and the tools and put them in the cloud so that it can access the data more easily. And so we'll be talking about these individual pieces more on Friday. But just to show like the important parts here, that we have these best practices whittles, it goes into a big repository of workflows, and then through that repository, you can access those workflows and apply them to your data in your workspace bucket, wherever that happens to be. This is some of the examples of our featured workspaces. Again, a lot of these have notebooks that we'll be going through in the hands-on sessions. For example, germline SNPs and indels. There's a notebook for joint discovery, or a workflow for joint discovery. Most of them have notebooks to go with them. And it's a beautiful system because then you can take that featured workspace that we have, you can copy it, you can add your own data, and then everything should be ready to go, and you can just change the inputs to point to your data and maybe some optional parameters that you may want to tweak to do your analysis in a slightly different way. But most of the work is there for you. The data will still point to our example data if you want to keep it that way. And all the parameters, like what filtering level and how sensitive do you want it to be, that's already part of the workflow that you don't need to worry about. So to give an overview of our schedule for the rest of the week, this morning, we're mostly going to be doing introduction presentations, talking about all kinds of variation, both on the germline and somatic side. And in the afternoon, Kate is going to walk you through a germline variant calling analysis with haplotype caller, genotype GVCFs. Day 2, we're going to talk in depth about our germline SNP and indel methods, both for calling and for filtering. And in the afternoon, I'll be doing a hands-on with you to do some hard filtering and also to use our new, shiny, latest and greatest convolutional neural network tool for deep learning and variant filtration. Day 3 is going to be devoted to somatic, talking about mostly our SNP, SNV and indel calling methods with some hands-on applications. And then day 4, we're going to talk a little bit about the rest of the ecosystem, running WDL scripts against Cromwell and using Docker potentially if your machine supports that at the moment. But at least you'll be looking at the scripts and actually working with them and modifying them. Any questions? Anything you want to make sure is covered that you don't see on the agenda? All right, let's dive right into it. So in order to talk about genomics, first we need to talk about where the data actually comes from. In this talk, we're going to talk about the front end of the pipeline from the actual samples, like the test tubes with blood, spit, whatever it is that you're deriving your DNA from, what happens on the sequencer, and what comes out of the sequencer and what that looks like. So the first step, once you have your DNA from whatever source of biological material you're going to use is library preparation, library prep. So starting from your sample of DNA, typically we run a couple rounds of PCR to be able to get more copies of all those DNA molecules that you're going to be using in your analysis. We will shear the DNA into fragments because, each chromosome as a single piece is a couple feet long, so it's very hard to work with. And we want to shear it into smaller fragments so that we can have those paired end reads that are so informative. And then there's a purification step for the fragments. This is when we would do size selection, because in order to take advantage of your paired reads, there's sort of an optimal distance that you want the first in the pair to be from the second. And then if you're doing hybrid selection, gene panels, then that's when you're going to add your baits to pull out specific parts of the DNA for coding regions that you're interested in, for example. And then we have these fragments. So typical fragments are about 300 base pair. And then that's our original source DNA. And we're going to add to it adapters, which are going to help the DNA actually bind to the flow cell, and primers, which is going to assist in the PCR replication that we're going to do, because we only sample a small proportion of the DNA molecules when we do the sequencing, and so we want to make sure that each of those is represented very well. So we make multiple copies of it with PCR. Often we will multiplex our libraries, which is to say that for each sample, we'll split it up into separate aliquots, separate subsamples, and mix it together with the other samples that we have in our study so that we can run data for each sample on multiple lanes across the flow cell, which makes us more robust to errors and particular flaws in the flow cell. And we'll see in base quality score recalibration why that's important. We run the amplification, and you probably have seen these videos where the little fragments, they bend over and they do the PCR, and someone in our group likes to call it the worms nodding their heads. And so this is all building the reads as sequencing by synthesis. So for each base in the DNA, which is effectively a position in your read, we flow the reagents with the 4 different colors over, well, usually 4 different colors, over our samples. And then inside the sequencer, the camera takes a picture. And so we know what each color is at each position. And so as we go through each position in the read and add more reagents and continue building our complementary DNA strand, then we have all these pictures with different colors, and from that we can derive the bases in the reads. And from that we get a big pile of reads, tens, hundreds of millions, maybe even more, depending on the type of experiment you're doing. So what does the output of the sequencer actually look like? Mostly it's going to be in a FASTQ format. And the format for that is for the first line in each description of the read to be the sequence name. In most of our applications, the read name will have important information like what library it came from, which lane on the flow cell it came from, which flow cell, even what position within that flow cell, which we use for some analyses. Then there'll be the actual sequence itself, like the A's and T's and C's and G's. The strand, which in our case we output always the positive strand. And then the associated quality scores with each of those bases. So the sequencer can estimate the quality of the bases based on how clear those images are effectively. As you continue to sequence, some of the molecules will fall behind in their synthesis and the color won't be as clear. And so we call that base with a little bit lower confidence. We say, oh, it looked pretty green, but it was kind of a little reddish maybe. And so that can be reflected in the quality scores in the FASTQ file. which is important information that we need for our variant calling later on, because we want to know how accurate each of those observations from the sequencer actually was. And we do this in the Fred scale, and as hard as I tried, I could not find out where the Fred name comes from, but it's scaled such that it's easy to represent very, very small probabilities as integers. And so mostly we're going to be working with things that are very close to 1, right? We want to have high confidence, 99% confidence with a 10% error rate, 1% error rate even lower. And so representing those small numbers of the error rates is a little bit easier if we do it in log 10 scale. And then because that's going to give us a negative number, we'll multiply it by negative 10 so that we have significant precision and it's easier to work with. So after working with these numbers for a while, you kind of have an intuitive use for them. But the numbers to remember are that Q10 is 90% confidence, Q20 is 1 in 100 errors, and Q30 is a 0.1% error rate. And so all those sound pretty good, but keep in mind that the human genome is 3.2 billion bases. And so if you have 0.1% errors, over 3.2 billion bases, that actually ends up being a lot of errors. Typically, Q20 is pretty good for most analyses. Certainly for base quality scores, it's very good. At variant calling, a lot of the times we'll use Q30 or even higher. So before you embark on your own sequencing experiment, the first important question is, do you want to do whole genome or exome and targeted capture? And both of them have pluses and minuses. Whole genome is obviously a lot more expensive. It's at least 3 times more expensive than a whole exome. But the good news is that the coverage is very even, which helps in copy number calling. And you have observations from all parts of the genome, not just the coding parts. So for structural variation, you're more likely to be able to capture the exact breakpoints of the event. And if you have any interesting variants that are intronic, then you'll be able to capture those as well. With the exome, you get even sometimes higher coverage than a genome would at the same position, but again, it's limited to just the targets that you selected, which are usually coding regions. So if there are variants of interest, like a new splice donor site that's being generated by a variant in the intron, then you'll be blind to that in exome sequencing. But it is more cost effective, and even though the coding regions are 1% of the genome, that is the set of regions that we know the most about. So a lot of analyses will use exomes. So this is a screenshot from IGV, the Integrative Genomics Viewer. We'll play with that a little bit later this afternoon. And just to show you that this is the whole genome observations where all the gray bars are reads, and we're showing the coverage track up at the top, sort of as a histogram. So for genomes, pretty consistent, pretty even over both the exon there shown at the bottom and its associated introns. For the exome, it's only having coverage with respect to that blue rectangle that's representing the exon for this particular gene. And so both of these BAMs are able to capture the SNP that's that orange and red bar kind of in the middle of the exon, but only the genome can capture with good confidence the insertion that's shown in purple that's farther into the intron. And then there's another SNP where the exome doesn't have any reads at all. If you choose to do exome sequencing analysis, different exome kits will produce slightly different data, which is important when you do copy number analysis, for example, because we want to make sure that the coverage is consistent across samples. So for example, here we're contrasting kit A on the top with kit B on the bottom. And then along the bottom, we have our gene model with the exon as the larger blue block on the right, and then the 3' untranslated region as the skinnier rectangle on the left. So kit A provides pretty good coverage for that exon. That looks good. Kit B has a lot of reads, but most of them are focused on that untranslated region that's not of particular interest, and then its coverage over the exon itself is not as good. So having even coverage over the targets that you want is an important part of evaluating your exome sequencing. So we're going to talk about quality control and how you know if your sequencing looks good and is good enough to use in an analysis. And so this is kind of looking forward a little bit into after we've done the alignment and after we've looked at the reads, so a little bit of a spoiler, but you do need to do some analysis on the reads to be able to get the metrics that you want to interpret to evaluate the quality of your data. So some of the bad things that can happen is that you have a normal number of reads, a number of bases, but the coverage of your targets themselves is actually poor, like in the previous example. Another bad thing that can happen is a high proportion of chimerism, which we'll define in a couple slides. A bad insert size distribution, too wide, too skinny, weird outliers, particular artifacts associated with oxidation, which can happen in exome sequencing, and also FFPE-based data for cancer samples. And another thing is that the library size could be too small, which you'll know because there's a high rate of duplication. Exomes have their own problems, unevenness in the coverage distribution like we saw in some of those screenshots. We can measure that with the Picard metrics, the fold 80 penalty, which we'll talk about in a little bit. And then a lot of those oxidation artifacts are more common in exomes than in genomes. In genomes, we can still have additional problems, like a high proportion of unmapped reads. Where are those data coming from if they're not coming from the human reference genome? Or a high percentage of adapter sequence. So if you have a bad insert size distribution and the reads start overlapping each other to the extent that they read past the beginning of your fragment into the adapter upstream, then you're losing a lot of data that you effectively paid for. So there are a lot of factors that influence what your coverage actually looks like. For example, in exome and whole genome sequencing, the coverage is going to be sensitive to what the GC content in the reference is. And so thinking back to your biochemistry, the nucleotides have hydrogen bonds between them, and A's and T's have 2 hydrogen bonds joining them, and C's and G's have 3 hydrogen bonds joining them. So as you go through PCR, and you do the melting phase, it requires more energy to break those 3 bonds. And so fragments that have more G's and C's are less likely to be replicated during PCR. And so we have this systematic bias that will have a little more coverage in the AT-rich regions and a little less coverage in the GC-rich regions, which is probably what happened with these samples here. Yes? So there are, well, PCR-free genomes do a lot better nowadays. We use exclusively PCR-free kits at the Broad nowadays. With the PCR plus kits that we used to use, they did have particular reagents that they would add to try and overcome that bias. Yeah, I don't know exactly what they were, but if you looked at the distribution of coverage by GC, it wasn't quite as bad as you would thought. PCR-free is definitely better, but they did as much as they could for the PCR plus to try and balance it out. Because sometimes you just don't have enough data, like you don't have enough DNA material to do PCR-free. So when we talk about exome coverage, just having a good average coverage doesn't tell you everything you need to know, because the distribution of that coverage is going to be important. So here we're showing 3 different hypothetical exome capture kits. Kit C just looks bad all around. These numbers are really, really tiny, but the maximum is 10, and that's just in some positions. So, I mean, 10 observations of a base is not great, but most of them are even lower than that, with like the beginning bases only have 1 read covering them, and so you only have 1 observation. So it's obviously bad. Kit A is going to have a really high mean coverage, which seems good, right? But this whole screenshot that we're showing, those are all coding bases of the exon. And so we want to have coverage over all those bases. And so even if we have a good mean coverage for KIT A, then the distribution is not very good. And so we'll measure that in the Picard hybrid selection metrics as the fold 80 penalty, which is how many times more coverage would you need to get 80% of the bases at this target up to the mean for the whole target. And so for example, KIT B has a pretty low average coverage, but how many times would you need to multiply that coverage to get the rest of these little poorly covered bases out there up to the average? I don't know, off the top of my head, maybe 3 or 4. But for example, KIT A, the average is going to be a lot higher, and the bases that are poorly covered are a lot lower. So you would need a lot more sequencing to get those poorly covered bases up to the average. So effectively, when we talk about fold 80 penalty values, lower numbers are better. Here's an example of a good-looking genome and a not-so-good-looking genome. The mean coverage for both of these is around 30, which is our typical genome target. The percent of bases at 15x for the top sample is only 69%. And if you think about what you want your distribution to look like, a lot of your bases should have at least half the target coverage, right? Half the goal coverage. So a more typical value would be 75%, like for the bottom sample, or even higher. But again, we're taking 2 different measurements here rather than just the mean. We want to represent the whole distribution of coverage with the mean and then the observation at a lower level of coverage. One of the things that can happen is that across our read groups, we might have varying levels of coverage. For example, read group 1 looks pretty good. I'm assuming this is an exome experiment because it's very uneven. Read group 2, we really didn't get a lot of data from that, unfortunately. But if read group 1 didn't meet our target by itself, we can always run another flow cell to effectively top up coverage, as we say, and generate a 3rd read group that will help us reach our coverage goal. So chimeras, sounds very exciting. These are reads where the 2 mates map very far away from each other. They could be on entirely different chromosomes, or they could just be 2 or 3 standard deviations beyond the mean insert size. Which is to say, like, you expected all those black templates that we had in our library prep slide, you expect them all to be about the same size, but if you have 2 reads that map really far away from each other, it either means that you had a really big DNA template, which seems unlikely, or something strange has happened in terms of maybe an actual structural variant event, or possibly something that happened during DNA extraction, where the DNA actually ligated to itself in different positions. So in IGV, we'll show these reads that have a mate that maps to a different chromosome or colored by that chromosome. This region looks pretty wacky. It's showing reads all over the genome. And then the ones with the rainbow bases are showing soft clips, which means that part of that read mapped in one place and part of that read mapped somewhere else. Yes? When we've seen samples with high levels of chimerism, we've generated new libraries for them, and it didn't help. So our hypothesis is that it does happen during the sample extraction. And the other sort of piece of circumstantial evidence is that when we do our own sample extraction, we don't see this nearly as often as when someone will send us their already extracted DNA. Yeah, so in these samples, you'll see an incredibly high percentage of chimeras. Like, structural variations are pretty rare in the genome. Let's see, on the order of like thousands for a single genome. And so the number of reads that represent thousands of breakpoints is going to be a very, very small percentage of your total reads, whereas this horrifically bad sample is showing 30% chimeric reads. Like a third of your data is mapping in a very strange way. Yeah, so we'll use data. 5% is okay, and then 10 to 15%, it kind of depends on what kind of analysis you're doing. For somatic, I would be stricter. For germline, we'll often pass samples that are up to 15% chimeras. And so when we talked about chimeras, I alluded to this insert size distribution. So for example, this is a whole genome sample, and when we did the size selection during our library prep, our goal was to have most of our fragments between 400 and 500 bases. We do 150 base pair reads for our germline whole genomes, and so that's a good amount of space, so we're not wasting too much coverage double counting bases with both reads or small fragments reading into the adapter. There's also, a significant proportion that are smaller than that, and that's okay. But then there's this enormous number of reads at this incredibly small insert size, which is a big red flag for us. The way that we discovered this sample in reality was because it had these big pileups of soft clips, and so our variant calling methods were calling a huge number of de novo mutations, because this was a family study, and the vast majority of them were insertions. So whereas usually we expect kind of a closer proportion of insertions to deletions. So we started looking at the data and we said, oh my goodness, there's clearly something wrong here. Because we have, if you look at the coverage track, all of the parts of the reads that are not soft clipped are very small. They're 17 bases, I think. And coincidentally, 17 bases is the, yeah. Hmm? No, but that does cause a different kind of artifact. 17 bases is the minimum exact match for the aligner. And so we took some of these reads in their full sequence, we ran them through BLAST online, and they started mapping to bacteria. So these particular samples were saliva samples from patients that were immunocompromised. And so a significant proportion of their DNA was bacterial. And the aligner did the best it could, and periodically it would find something in the bacterial genome where a little bit looked like human. It's okay, I'll try and put it there. But for the most part, most of the DNA was unmapped. So we had a large number of unmapped reads as well, which is a warning sign. So we ended up putting in a filter in our early BAM pre-processing that will flag reads like this so that they're not used in subsequent variant calling. We don't throw them out because we don't want to exclude the possibility of weird structural variations, but at least they won't be shown in your IGV pileup, they won't be used for your insertion calling, and they shouldn't be generating a lot of de novo mutations. All right, so we talked about what happens between your biological sample until you get reads out of the sequencer. And then next, John is going to talk about what we do to pre-process those reads before we do variant calling. Are there any other sequencing data questions?
All right, so now that we've heard a little bit about how we get our data and what the sequencers are actually doing, I'm going to talk a little bit more about, now that we've got our reads, how do we pre-process them so that we can most effectively call variants off of our data. Okay, I'm going to do that then. All right. So, Laura just told us a little bit about the sequencing at the beginning, and like I said, I'm going to talk about what it takes to go from the reads to going into our best practices pipeline. And briefly, it's 3 steps. It's mapping the reads to the reference, making sure we account for any duplicates that have occurred in our reads, and then recalibrating each of our bases so that we have a very good estimate of what the base quality was for each of the bases inside each of the reads. And all of these things are very important when it comes to the assumptions that we make and the kinds of algorithms we use when we do our variant calling itself. All right, well, that's okay. So, briefly, the data that we're getting from our sequencers themselves is basically a whole bunch of small segments of the genome of whatever sample we have that is not aligned to anything. We just have a whole bunch of bases that come together, and we then need to make sense of them. So, without the mapping information, we can't do any analysis, so that's one of the first steps we're going to do. The other problems that we have are some of these sequencing systems have different kinds of error modes. Now, depending on what you're working with, these error modes can be pretty straightforward and pretty simple, and they can be known ahead of time. Other things are a little bit more hard to tell, like when the sequencer tells you what it thinks the error rate is and the actual error rate is different. So we need to account for that as well. And there are other technical artifacts that can occur either in library prep or in the sequencing process itself that we need to account for. So briefly, in summary, we need to map, then sort, and clean up the reads so that we can use them. Yeah. So, like I said, we have a whole bunch of small segments of the genome that we don't know where they should go in terms of our reference. So the first thing that we're going to do is we're going to map those segments to whatever reference we're using. When we're doing mapping for DNA, we use BWA, the Burrows-Wheeler alignment algorithm, and we use STAR for RNA-seq, and I'll get into a couple of the reasons why we do that a little bit later. But the mapping process will take all of these reads where we've got a whole bunch of data here and here and here and here, and they'll align them to the reference so that all of the segments that we know about align to regions that they should align to. And so the goal is pretty straightforward. It's to, from our sample, make sure that we line up all of the segments to the regions in the reference that they actually occur in. And sometimes we're going to have SNPs or indels and other variants that occur in our data set that don't occur in the reference, and the aligner has to be able to be robust to these errors so that we can identify them later as variants. You'll see that in our sample itself, we've got a couple of SNPs or indels, and we actually have a full duplicated region here that corresponds to this second region. Now, if we could read the molecule from start to finish in one go, this would be relatively straightforward because we would know where everything is, but we can't do that. Instead, we have a whole bunch of small pieces. So the problem is a little bit more difficult. We have all of these short reads that in this first case, it's pretty straightforward. All of the regions align the way you would expect them to, and some of that is helped by the fact that reads from this region align here, and we know that this other part can align here. But there are other cases where it's not so obvious. Like we saw in our previous sample, we have a duplication of that green region, and because of that, the data is doubled in our sample, and so we don't really know which part of this green region each of these reads comes from. These on their own could align to either one. And one of the ways that we can deal with this is with paired end sequencing. Now what that means is each of our reads has, we get 150 to 300 bases from next generation sequencing platforms. And the paired ends sequencing is on the molecule itself, we get a bunch of data from one end and we get a bunch of data from the other end of the molecule, and we know that those two things correspond together. There's a gap in the middle, but we know how big the gap is, so we know the relative spacing between these two pieces that we've actually sequenced. And because of that, even though we don't know what the middle data are, we can line them up and process them so that we know where each end occurs, and therefore we know that they are from one molecule and that those things are contiguous. So that helps a lot with indels in particular and other small to medium size variants and things like that. For very large structural events, the paired end sequencing is going to result in some strange mappings where one paired end will map to, say, a location on chromosome 1, and the other end could occur someplace completely different because you had a structural event in between the two ends. And that's one way we can do SV detection, which we'll hear about a little bit later. So, what do these reads actually look like once they come off the sequencer as far as the data goes? Well, we like to keep all of our data together, and we like to keep metadata as well. In the SAM or BAM format, which is what we use to store our data, we've got a header that tells us a little bit about the information for the sample itself, when it was sequenced, what the ID numbers for the machine were, what the read groups are, or what the version of the file is. And then each record has specific pieces of information as well. We have the read name, which usually corresponds to information from the flow cell itself, like the flow cell name, the coordinates on the flow cell, things like that. We have the flags, which will tell you a little bit of metadata about the read, whether it's mapped, whether it's unmapped, if it's a paired read, if it's a duplicate, which we'll talk about in a minute. We have the mapped position of the read, the quality of the mapping, which is, again, in Fred scale, with a cutoff on it. At 60, above 60, we don't report, because 1 in a million is pretty good, at least for alignments. We have the alignment cigar string, which will tell us how specifically that read aligns to the reference from that position. We have some information about the mate for the read, for paired reads. Then we have the sequence itself, the Fred quality scores for the bases, and some metadata. For instance, we see the read group here is read group A. And like I said, we like to keep our data and our metadata together because more often than not, if there's a problem, we need to go back and we need to see, what was the flow cell? Okay, well, when was it run? What were these different things that could have occurred that would contribute to an error? And when you're processing a lot of data sets, it becomes more and more important to have all this stuff together rather than separate, because then you don't have to look up one thing in another place and it reduces the possibility for more errors. So to jump into the cigar string a little bit, because it is pretty important, it's an acronym that stands for Concise Idiosyncratic Gapped Alignment Report. I have to believe that someone wanted to call it cigar and then named it that. But that's what it is, and that's what we call it. And for this particular read, you could see that the cigar string is, there's one S, 3 M's, 1 D, and so forth and so on. So if we look at what the reference is and the position of the alignment here, S is soft clipping, which means we keep the bases, but we don't consider them for being part of the sequence. For whatever reason, at the beginning, at the end, and the end of the reads, Sometimes we find that the reads don't map properly to the genome, and often it's because it's part of the adapter sequence or some other artifacts that have occurred at the very ends. And then once we get into the sequencing, things start to match a little bit better. The M operator is for matches or mismatches. So because we're doing alignment and we want to detect things like SNPs, we need to be robust to whether the bases actually are equal or whether they are different. There is another specific operator for base equality, which isn't shown here. But this allows us to align reads together in a way that is able to capture the spacing of the reads without worrying about whether or not there are actual errors. Then there are D and I operators, which are for deletions and insertions respectively. Like you would expect, D is for deletion, so we have a deletion in the read at this position, and we have an insertion in the read at this position before 8. And then another match at the end. This is very useful when we want to think about, at a high level, how well each read actually aligns and maps to a position, and whether there are any differences between the read and the reference where they occur. So I talked about keeping all of our metadata and our data together, but in practice, what usually happens is the sequencer will produce a FASTQ file, and the FASTQ has a bunch of raw data in it for your reads, but it doesn't have the ability to store the metadata that goes along with the reads. It can store base qualities and read names, but that's more or less it. So if we want to have something like a read group in the FASTQ file, we need to do something else. And the something else that we do is we take the FASTQ file and we actually process it into an unmapped BAM file. So it contains all the information you'd find in a BAM, except instead of mapping positions, we have another placeholder where there is no position. And that flags section in each of the data rows that I showed you before, one of the flags is whether the read is unmapped. So that's one of the ways we keep track of it. So we actually go back to an unmapped BAM, and we do the alignment with the file, the FASTQ file, and then that produces a mapped BAM file, and then we merge them back together with this tool called Merge BAM Alignment, which combines the raw data and the metadata into a nice cleaned and mapped and sorted BAM file that has all of the information we need from our sample. And in this way, we have all of our data together in one place so we can reference it later. So I talked a little bit about how we use a different aligner for RNA data. And the reason we do that is because of intronic and exonic segments. So if we're looking at the DNA and we're aligning to DNA for the RNA sequencing, we'll notice that obviously the introns aren't going to be in the RNA that we've sequenced. And because of that, you end up with not only these gaps, but they're replaced with ideally these N bases, which is another cigar operator that allows any base to be there. It doesn't know if it's a match or a mismatch or a deletion or an insertion. There's something there, and it could be anything. And in order to deal with this properly and be able to actually do good analyses, the aligner that we use is the star aligner. And that is able to better take into account the introns and exons in RNA data. So now that we've got a nice mapped file, we have to worry about duplication of each of the reads. Now, the duplicates that I'm talking about, I'll go into a couple of cases on the next slide, but the algorithms we use to do variant discovery, a lot of them depend on, I'll say, coverage of a particular base. So if you artificially increase the number of reads in a particular position, you're going to artificially increase the amount of evidence you have there for a variant being there, or for a variant not being there, in the case that a read without the variant is the one that's duplicated. And to account for those duplications and to make it so that our algorithms work a little bit better, we mark the duplicates with this mark duplicates tool that then will take all of these duplicated reads with that snip and only take one representative from those duplicated reads to actually use in the data set. Similarly, these reads down here are all compressed, or are all examined together, and one good representative is chosen from them because they're duplicate reads. Now you might be wondering, how do we actually, well, I'll get to that in a minute. So what kind of duplicates am I talking about? Well, we talked a little bit about PCR already, so if you're using PCR, then the more PCR cycles that you have, the more likely it is that a segment will be duplicated. And similarly, for the machines themselves, they effectively work as tiny cameras. And the way that the samples get plated onto the flow cells is such that they occur in tiny little colonies that get built up and built up and built up through sequencing, for the sequencing by synthesis process. So you grow a whole bunch of those worms that Laura talked about, the DNA worms or the RNA worms, and then you flow the different fluorescent bases over it and take a picture. Well, if for some reason when you were growing these little colonies of each read, one jumped somewhere or one colony grew too big, then when you're quantizing the image, two areas that are next to each other, if it's one colony of these reads, could appear as, if it's one big colony, those two areas could occur and look like two separate reads, even though it's just one big blob. And that's more or less how optical duplicates occur. And so for these two different kinds of duplicates, we need to be able to account for them in order to do processing on the data. So how do we determine duplicates? Well, we don't actually look at any of the bases. We look at the alignment position of the bases on the 5' end. And we look at the unclipped position. So I talked about soft clipping a little bit earlier, where for whatever reason the beginning of the read or the end of the read doesn't map very well to the reference. Well, we still take into account those reads when it comes to those bases when it comes to looking at duplicates, but only the position of those bases, not the bases themselves. So, for example, we're looking at these bases here, or these reads here, and all of them seem to start at the same position. These gray bases are the soft clip bases. Now when we have paired reads, some sequencers don't actually produce paired reads. We do the same thing on the 5' end of the mate and use both 5' positions as the key for whether they're duplicates. But in essence, we go through and we look at what the 5' position is of each of the reads, and then if they are all the same, taking into account the soft clipped portions, then what we do is we look at the base quality scores of each of the reads so that we can determine what the best representative read for that position is, and we choose that one, the one with the best scores. So, like I said, we like to keep our data and our metadata, and when we actually do the duplicate marking, we don't remove them from the data set. And this is so that if something goes wrong or if we want to take a look at those data later, we have a chance to do so. So instead, like I said, we mark the duplicates and add them as add the duplicate flag to the SAM file and then move on. Then if we want to look at what all the duplicates are later in the pipeline, we can do it after some analysis. And most of our downstream tools look at what the flags are for each read and then ignore them. So this is a pile-up view in IGV, and on the left we've got it configured to show duplicates. So you've got a whole bunch of these reads here, some of which are duplicates. And then if we turn off viewing duplicates, then you could see there's significantly fewer reads that are not duplicated. So one of the things that we used to do, we don't do this anymore, is we used to do local realignment around indels. Now this was because we had a strict pile-up based variant discovery algorithm that would make a pile-up, and because you're creating a pile-up and looking at each base together on all of the reads of those positions, you needed to take a look at them properly aligned to each other, and indels would cause a shift in that position. So before we had to use an indel realigner that would realign the reads locally and then do variant discovery. Now, in haplotype caller and MUTEC2, the algorithms on their own already will do local realignment around variants that they're looking at. So we don't have to do this anymore, but it's something that used to be in our pipeline.
What's the tolerance for mismatches and exact duplicates? So they have to have exactly the same sequence, or they can have like some mismatches?
Yeah, so the duplicates actually don't look at the sequence information at all, which sounds a little bit strange, but it looks at the position that the sequence starts. And so it's, in terms of figuring out which reads are duplicates, it doesn't matter which, it doesn't matter what the quality of the bases are in and of themselves to determine that there are duplicates. But once you've decided that, you know, these 4 reads are all duplicates together, then you would look at the quality to figure out which one of those reads you want to use as the one read for that position. Does that make sense?
Yeah, in terms of quality, but in terms of sequence itself, I mean, if you have one mismatch in the, let's say, 100, you would consider a duplicate only with one mismatch, or if you have 4, then it's not a duplicate? Or what's the criteria for telling it's a duplicate or not in terms of sequence mismatches?
Right.
So what you're doing is you're trying to weigh the probability of having a duplicate that also had a sequencing error in it versus having 2 DNA fragments with exactly the same start, like exactly the same DNA fragment, like chopped up during sonication and extracted during size selection. And it's a lot more likely that it was a PCR duplicate with a sequencing error rather than 2 of the exact same molecule.
So basically, you just know that it's a duplicate because it starts from the same position, and then you choose which one you'll use based on quality.
Yes. Yeah, and again, it's because, like Laura said, it's pretty unlikely that you would have these tiny fragments all starting at the same position together when you're actually breaking up the DNA. Yeah. So now we've mapped our reads, we've marked our duplicates, and we've got a whole bunch of reads that are probably good. But we don't know how good the base qualities are for each of these reads, because even though the machine will tell us that the quality is X, the quality could be very different from that. And so we have a tool called Base Quality Score Recalibrator that will look at what the expected quality for each of the bases are in each of our reads, and look at what the empirical quality is by calculating whether there are mismatches or insertions or deletions between the read and the reference that we would not expect, and then adjusting the base qualities in the right direction to make them correct empirically. So, for example, we have the machine, the sequencer itself will report certain qualities for each of the bases, and we can then calculate the quality that we see for the reads in each of the bases as well. And if we look at this plot, the purple dots are the original qualities that are reported by the sequencer. And if the reported quality is the same as the empirical quality, you would expect it to be aligned with a slope of 1. It would go up the diagonal. But what we see is it's a little bit lower than that. And so for each of the reads, we look at what these differences in quality are based on several different covariates. And we adjust the reads based on those covariates to then, for the qualities, align back up to that diagonal line. As a specific example, one of the covariates we look at is dinucleotide context. And we have a similar graph over here where we've got each dinucleotide pair and then the accuracy of the quality score for each of those pairs. So if you've got an A, then it's, you know, a little bit below what we would expect, which is the 0 line. If it's an A, then a G, then the quality is worse. And for CG, it's as bad, it's the worst of any of them. And interestingly, we overestimate the quality for TA, or at least when I say we, the sequencer overestimates the quality. But because we know that the empirical and the reported quality should be the same, we're then able to adjust the quality for each of them after reading through all the reads and seeing what the actual quality scores are. There are other covariates that we look at as well to adjust the qualities, but this is one of the prime examples because of the big differences that we can see in the CG quality and the TA quality there. But then how do we calculate this empirical quality in the first place? Well, like I said, we go through the reads and we look at whether there are insertions, deletions, or mismatches between the reads themselves and the base, the reads themselves and the reference. And it's a pretty straightforward calculation. The only caveat here is that we ignore known variants. So if we expect that, you know, at position X on chromosome, or position Z on chromosome 5, there's a SNP that's an A to a T, then we'll ignore that if that occurs in our data so that we don't count that known variant against the quality scores that we see. And then we throw in the number of mismatches to the reference over the number of observed bases or the length of the read. And we get a probability that we then convert into the Phred scaled quality score for reasons that Laura talked about earlier. And if you're wondering, the 1 and the 2 here are so that when we observe no bases, we have a 50-50 shot of being right. Well, a 50-50 shot of the quality being good. And also, one other thing is we use a few different data sources for the known variants for the samples that we sequence. If you don't have known variants, then you can bootstrap it by running a variant discovery algorithm like haplotype color or mutect, and then using those variants on your reads to reprocess them with known variants in their place. Like if you're sequencing a novel organism or you don't have a good baseline set of known variants. And so this is what a recalibrated SAM file looks like for one read. This would be the end of the line of the SAM record. But we have the recalibrated base qualities that now occur as the regular base qualities in the file. And we save the old ones under this SAM tag later as some metadata in the file. Again, we like to save our data so that we can go back and take a look at it later if there are issues or if it's something we want to do for our study. And so that's the short version of our read pre-processing that we do after we get the data from the sequencers and before we start doing variant discovery on them. Next, we have a short break, and then we'll start talking about some variant discovery techniques and other algorithms in our data.
tend to recommend getting to a max, but one of the errors that people were hitting involved the Docker not starting, and my team told me that to try restarting the computer, and that's the feedback they found from Docker's support section. Try that. If it doesn't work, let me know your error message. I'm going to continue working on this, but we won't need Docker till day 4.
You were talking about the error in some Docker with the Windows combination, or?
Yes.
Are you using a legacy version of Docker? So they told you to just restart everything before?
Yes. I'm suspicious, but if it works, it works.
Well, I tried to restart it and it gave me the same error I had yesterday.
What is
The only error message that gave me is that there was an error in the installation. Because they say in instructions that the version that you sent us is not compatible with Windows combination, it needs to be Windows Pro or so they want me to make those design correctly and install a legacy version. But I noticed that it was installed.
Ya vamos a iniciar, entonces para prestar atención a la presentadora.
¿Listos? Bueno. ¿Todos listos? Bueno. So now we're going to talk about variants. Now that we have our reads, what do we do with them? I'm going to give you an overview of all of the tools that we have in GATK for discovering all kinds of variants. So just to give you an overview, there are 3.2 billion sites in the human genome, but any 2 people share about 99.5% of their DNA, which means that we can describe the genome most efficiently as the very few places where it's different from somebody else. And for that reason, we choose a single human reference genome, and then we can very efficiently describe any other person as their variants from that reference genome. Just sort of like a little population genetics brain teaser. Any person has mostly common variants, variants that you've inherited from your parents' de novos and their parents' de novos and so on and so forth. But as you start sequencing populations, most of the variants that you're going to see in the call set are rare. So, talk more about the reference. From the conclusion, I suppose, of the Human Genome Project in 2003, that gave us HG16, which is the almost complete assembly. And from there on, we worked on getting all of the contigs mapped to chromosomes and figuring out how all the little pieces fit together, because as we do the assembly, there end up being more chunks than there are chromosomes, and it takes a little while to figure out exactly where they belong. But now at GRCH 38, which was released in 2013, we think we have the human reference pretty much as good as we're going to get. The cool features of that new reference are that it has these alternate contigs. Well, it has extensive HLA sequences, which if you're interested in that, it better represents all the diversity that you'll see in that particular region which is relevant to the immune system. And there are also alternate contigs, which better describe other populations. And so on the plus side, we're able to capture more variation, larger variation that we would see. But on the minus side, it's going to cause some complications with mapping. Just to tell you a little story that never ceases to amuse me. The UK Biobank project was sequenced not by the Broad, but by Regeneron. And they did not appropriately account for the alternate contigs in the H238 reference. And they have a whole slew of mapping quality 0 reads. And someone published a bioRxiv preprint talking about the thousands of missing variants from the call set. Whereas to do our own internal analysis, we reprocessed our data, we realigned it, we used our own duplicate marking, and so on and so forth. And other benefits, in H238, they generated effectively synthetic sequences for around the centromeres, because those satellite repeats can be very complex and confusing. Most people don't really use them as far as I know. And then there's a masked version of the reference which will exclude low complexity regions, which makes some types of analyses a little bit easier. So here's an overview of all the different types of genomic variants. We typically divide things up into germline versus somatic variants, and germline obviously are things that you're born with. They came from the germ cells of your parents. Not to say that you can't have de novo mutations there, but all of your cells will reflect all of the same germline variants for the most part. Somatic mutations are those that arise later on in your life, typically in a single cell of your body, not necessarily of the germline. And then from there, it may grow into a larger population. But we use different variant calling methods for the two types of variants, and we'll go into more detail about that a little bit later. But both for germline and somatic, we have short variants, which we typically kind of cut the bounds of the short variants at 50 base pairs, and then once you get into maybe 200 base pairs, it becomes a structural variant. It's kind of arbitrary, but mostly it has to do with the size of the variants that the tools are able to output. Haplotype caller definitely does the best for things that are less than a read length. In IGV, we saw some of the screenshots. You'll see deletions represented similarly. Insertions have a little purple, like caret, like you would start a text box. And then beyond 50 bases, we get into structural variations, of which copy number is one. Sometimes we talk about copy numbers separately because exomes only are able to represent that type of structural variation. So there's, a pretty strong depth-based signal for copy number variants. For homozygous deletions, you'll see no reads. For heterozygous deletions, your coverage will drop by about half. And there's also copy gain, duplications, and so on and so forth, where you'll see a higher coverage than average. There's some other signals that we can use in SV calling as well, which I don't think we'll go into today, but like we talked a little bit about when we were talking about chimeras and insert size distributions, the other SV signals are places where the reads tend to map either really far apart or unexpectedly close together, places where they change orientation, maybe the two reads in the pair are pointing out. And then, of course, the most obvious are these breakpoints, like we see here between the orange chromosome 1 and the purple chromosome 5. All of those are reads or pairs of reads that map to two different chromosomes. In this case, we would call it a translocation. Like part of the DNA of chromosome 1 was translocated onto chromosome 5. And those will be often represented in IGV with those like rainbow colored soft clips that we saw earlier. We also have a tool in the GATK for pathogen detection and classification. We call it PathSeq. It's the latest incarnation of a tool that was started in the Broad's Cancer Genome Analysis Group. And honestly, that would have been really useful when we had done that analysis with those little 17 base giant pileups of the bacterial reads, because there we take all the unmapped reads and align them against a reference microbial genome, or a reference composed of multiple microbial genomes, so that we can estimate the abundance of all the different species. So here's an IGV screenshot just to show you what exactly these are going to look like. We'll see the insertions in purple on the left, the SNPs. Here we're showing both the reference base and the alternate base are colored, and they'll show up in the pileup up at the top. So as you're scanning through really quickly, it becomes obvious. And then here we have those rainbow soft clips. And they're not in good agreement with each other, honestly. Like if you look, some of them are, like these look pretty good, but if you look up and down that whole column, or if you look at the pileup here, there's not a good balance of two different alleles. So I actually am not sure that this is a SV breakpoint. I don't think it's consistent enough. I think it might be instead some other kind of error mode. But it's a signal similar to that you would look for. And then the reference genome is given at the bottom. IGV will take like a refseq file for gene models, so you can tell if you're in an intron, if you're in an exon, if you're in UTR. And so that can be very useful as you're interpreting your variants. The file format for variants is called our variant call format. And it starts with a header, which is particularly useful because it'll tell you what version of the file format is being used, typically what the reference is that it was aligned to, which as we upgrade all of our pipelines and we start using newer references, it becomes more important that we do all of our analysis with all of our resources and data on the same reference. In the header, we also define any annotations that we might have in the info field or the format field. So for example, in the info field, we have AF, which is allele frequency. The info field annotations are going to represent all the samples in aggregate. So that's going to be the allele frequency over all three of the samples, for example, in this VCF. Whereas if you saw AF in the format field, which I think we'll probably see in our somatic variant calling talk, that will likely be defined in the header as allele fraction. And because that one appears in the format field, all the attributes in the format field apply to the specific sample. So for example, in the germline, in this example, we don't have a lot of data per sample, but there's some metadata there to help you assess the quality of your genotype calls. The VCF format does also support copy number variants and structural variants. It starts to get a little hairy depending on how complex the variant is. But for example, if you're running our copy number tools, then in the alternate allele, you'll see deletion or duplication, because rather than giving the full sequence that was deleted or duplicated, which could be hundreds or thousands of bases, we give it a little symbolic allele. Sometimes if it's a smaller event, there'll be a consensus sequence in the info field. And then we give other information like the length of the SV. So knowing that it's a deletion, what position it starts at and its length, if you really wanted that consensus sequence, you could figure it out using the reference. There's also a confidence interval for the start position and the end position. This is because a lot of structural variation occurs in repetitive sequences. It's just part of how the genome crosses over during meiosis. And so within those repetitive sequences, sometimes it's a little hard to localize where the breakpoint is. Is it in the first TAC? Is it in the 23rd TAC? So we try and give an estimate of where we think it is. So again, we have workflows for most variation types, both on the germline and somatic side. Like I said, I hope within a couple weeks we should have something public to offer for structural variation, although we really haven't started work on that on the somatic side yet. So we're going to start with germline SNPs and indels because they're the easiest. And we talk about germline SNP and indel calling in the context of joint calling, joint discovery, joint analysis, because looking at a single genome is not really that useful. It does tell you how your sample is different from the reference sample. It doesn't tell you how your sample is different from other samples in that population. It doesn't tell you how common these differences in your sample are. Like if you're doing rare disease research, whatever the causal variant is, you expect it to be something that's incredibly rare. Otherwise, everyone would have that disease that had that variant. So being able to analyze these genomes in the context of more samples gives us a lot more information. So as of a long time ago, we got to the point with our germline SNP and indel cohorts that we couldn't let haplotype caller process all the BAMs at the same time anymore. It was just way too much data. It was too many file handles. There were like, operating system challenges. There were compute challenges, memory challenges, and everything fell apart. So we moved to a system where we do the variant calling on each sample individually, and then we save some additional data so that when we combine it together with other samples, we can still represent the allele frequency across all those samples accurately. So this is an example of a genomic VCF or GVCF. We call it a genomic VCF because we give information about every position in the genome. So at the top, the blue lines are giving our reference confidence blocks, which we'll talk a little bit more about in the haplotype caller talk. But just to show that there's additional data being represented in the GVCF, and then when we combine our samples together, as in the bottom, we'll see that those non-refs are gone, and we have and then the reference blocks are gone, and we have just our confident genotype calls. After we do our variant discovery, we have to do filtering because haplotype caller's responsibility is to be as sensitive as possible so that you don't miss anything. And you can filter it out down the road, and that's fine. But we want to make sure that we can get all the variant information we can, because at that point we're going to throw the reads away and not look at them for the rest of the pipeline. So given that we want it to be as sensitive as possible, admittedly there are going to be some false positives in the haplotype caller results. So we pass it on to variant filtering. Variant filtering can be done by hand, and we'll do a little exercise this afternoon where we do some of that. But it's a lot more complicated than one can typically draw good boundaries in this multi-dimensional space because we use many different features of the sequencing. And so trying to divide that space into good things and bad things becomes increasingly complicated. And you can see that as we project this into 2 dimensions, those distributions of good pass variants and bad variants that are filtered out with the VQSR tranche labels, they overlap pretty significantly in these 2 dimensions. And so it's very hard to visualize it as a human being in higher dimensions, but when we reduce it down, then you lose a lot of that specificity. So that's why we have VQSR and the CNN model, is that it can better grasp the multidimensional data and use it efficiently. Finally, we recommend that people end their pipeline with genotype refinement. So after we have our recalibrated variants, those are really just genotype likelihoods. It's just information about the genotype based on the reads that we have. But that's not all the information that we have in the vast majority of cases. In almost any case, we have population information. For example, if you see a variant at a location where you have never ever seen a variant in any human being before, is that suspicious? Perhaps so. So maybe we should require a little bit more evidence before we're confident in that variant. And so we can apply prior probabilities on the existence of variants based on population information. And we'll talk tomorrow about mathematically how we do that. And you might also have information about the family. If you're doing like a trio study, then you can use the information about the parents' calls and the offspring's calls to better refine the genotypes for all the samples involved. So this is an example from data that I worked on a long time ago from a trio of exomes. So we started out with 417 de novo calls from the filtered variants. And again, for an exome, The reported rate of de novo mutations kind of varies, and especially depending on the paternal age and so on and so forth, but that's way too many for an exome. For an exome, we typically expect maybe 2, probably less than 2, maybe less than 1. So 417, way too high. But then we can apply population priors, and we can use the family information to better refine our genotype posteriors. And then we have 17 de novo mutations at that point, which is looking a lot better. And then if we restrict that just to our highest confidence genotypes, then we get down to 8 variants. And 8 variants is probably still a little bit high, but at least it's a number that you could conceivably review manually. If you're working in a clinical context, then you can look at 8 different variants and potentially 8 different genes and look at IGV in 8 different regions, and that's perfectly reasonable, whereas 417 just does not scale. And then
How is the cutoff of the GQ filtering? How is the estimate amount of GQ score?
yeah, so we give the exact parameters in the talk tomorrow, but the high confidence is a GQ 30 on the posterior probability. And then the pipeline that we have will also output a low confidence if it's at least GQ10. And then if it's lower than GQ10, then it gets filtered out. So in the scenario where you're doing your clinical analysis, then you want to know, what do these variants do? And so to find that out, we run functional annotation. And this is using some sort of gene models like RefSeq, likely with multiple transcripts, to figure out, based on the reference context around this variant and how it changes with the variant, is this a synonymous mutation? Is it a non-synonymous mutation? Where in the gene does this occur? As you can see, the vast majority of variants occur in the introns. Introns actually take up a huge proportion of the genome, which is strange because they're not coding. But it's much more than you would expect, and that's why it accounts for such a high proportion of the annotated variants here. It'll also tell you about variants that are upstream or downstream. There's a lot of variants that are intergenic that are not coding, and we don't really know what to do with them. And then once we get into the coding variants, then there's a very, very small proportion of the variants in that sample that will actually make a difference biologically. So our overall end-to-end pipeline, which we'll cover a little bit today and a little bit tomorrow, we start with our recalibrated analysis-ready reads. We run haplotype caller to get our GVCFs. We combine them together across samples, joint call them. We do our SNP and indel filtering, genotype refinement, and functional annotation, and then we have an analysis-ready VCF. And then tomorrow, another one of the topics is going to be, how do you actually evaluate that call set? How do you know that your results are good? Especially because you're looking at data no one's looked at before. So what kind of metrics can we use to determine if we did a good job? And then this is an overview of the somatic side of the pipeline, which is for day 3. It's very, very similar with slightly different filtering steps. And then there's a little bit, a couple more resources that we use. For example, we'll talk about the panel of normals and some of the read information that we collect with another tool. So just to talk more about our somatic process today, give you an overview of what we'll be doing later. Somatic calling is a lot harder than germline calling, and it's hard for a variety of reasons. One of those reasons is that the tissue that you're looking at is very heterogeneous. So in this figure, we have nice normal-looking cells that are kind of yellow, beige, whatever. And then as they start to acquire mutations, they have behaviors that move towards being cancerous, and I guess we call them dysplastic. And then there'll be some subset of cells that replicate out of control and exhibit the other hallmarks of cancer cells, like, for example, in our blue population. But when a doctor, a histologist, goes to actually take that cancer sample, they have to take their little sample or scalpel and extract cells from this big mass. And so you're going to get some of the cells that you want, some of the cells that are still useful, and some cells that you don't want. And so there's a lot less signal in the somatic variant calling, which is what makes it hard. So from the germline side, we know we expect things to be at 50% for a het in diploid humans, or 100% if it's homozygous. There's a little bit of noise, some sequencing error, some mismapping, it's fine. But in the somatic side, we can't make any assumptions about what proportion of alternate alleles is reasonable. Because there could be contamination from another sample, there could be contamination from the normal in the tumor sample, which is going to dilute your signal. There could be a heterogeneity, seeing multiple subclones, some of which have the mutation you're looking at, some of which don't. And all of that adds to effectively the noise that you're going to see in somatic sequencing that makes the signal that much harder to pull out. So one of the things that we can do to get rid of some of the variants that we know we're not interested in is to use a matched normal sample. Typically we recommend blood normal, and we can use that sample to remove any germline variants that are going to show up in our tumor. Not that they're not real variants, but they're not real somatic variants. Because they're in every single cell and not all of your cells are cancerous, we expect that they're not the causal variants. So we can start to filter those out by comparing them with variants that are called more like a germline variant caller with a diploid assumption. And then what remains are the variants that are more likely to be somatic. And we can also use a panel of normals to help filter out commonly seen artifacts from our data. For example, John talked about how different reference context sequences like ATs are more likely to be errorful, or CGs are better. Those are things that you're going to see not just in your sample, but in other samples as well, although it may be with a low probability. So across many samples, you're more likely to see it. There may be things like mismapping reads from a large section of genome that's unique to that sample or unique to a small subpopulation. And those mismapping reads can generate a little bit of signal that looks pretty promising, but is not actually representative of the somatic mutations that we want. So, for example, we already filtered out the germline events that were in our tumor based on the calls in the normal, but periodically you'll have these artifacts, and they'll be a kind of a low allele fraction. They'll look maybe not great, but respectable. And maybe they don't show up in your matched normal for whatever reason. Maybe there was something different about the library prep that one day, or maybe it's just a low probability artifact and you got unlucky. So by adding the panel of normals, we're more likely to capture common artifacts that are going to be represented in our tumor, even if they weren't represented in the matched normal. So, yeah.
So when you say matched normal, it's the same person?
The same individual.
To see which ones are like germline mutations. But here it's a panel. It's a group of normal samples? Or?
Yeah, so it's ideally, and we'll talk about the requirements a little bit more on Thursday, but yeah, it is a group of samples, ideally from the same sequencing center, from the same type of study, hopefully healthy people that don't have blood-borne cancers. And sure, it's also useful to have their germline variants, but mostly we're looking for sequencing artifacts that are, because anything that we see that is low allele fraction in a healthy person, we're going to say is probably more attributable to an artifact than it is to an actual cancer driver mutation. So to do our somatic variant calling, we use our matched normal, we use our panel of normals, we use population frequencies for germline variants, and all this additional information goes into the calling because again, there's a lot less noise, and so we need more data to be able to extract the signal from the noise. And then once we have our analysis ready variants, we'll run Funcatator, which is John's baby, and we'll get our functionally annotated variants that we can use for analysis. We'll also talk about the somatic copy number workflow, also on Thursday with a hands-on tutorial. So again, as we saw in the figure early on, the hallmark of a copy number variation is this change in depth. If you have a normal exon in a normal genome, you expect it to have some coverage, and then if you see coverage that's significantly elevated beyond that, then you might expect that it's a copy number variant. The other thing here that's a strong signal is this red SNP that's at 80% allele fraction. Granted, with only one reference read, you can't be that confident, but seeing variants that are at allele fractions that are not the 50% and 100% that we expect is another signal that there might be something going on. So the basic steps in our somatic copy number alteration pipeline are to collect read counts. So we're effectively just going to do coverage like our IGV track pretty much, although we're going to do it in bins because we need to reduce the amount of data to be able to run these algorithms. So we can't actually look at each base pair, but we look at bins either for exon targets or maybe 150 base bins for genomes. We're going to normalize those read counts across samples for the same target and across targets to account for samples that might have slightly higher coverage because they went through a read group top-off like we talked about. And then once we have our normalized depth signal, we can segment it into events because we expect that a lot of these events are going to cover multiple exons, so we'll see multiple observations that are of elevated copy number or depleted copy number, and we want to know what are the boundaries of that single copy number event. So there's a variety of different steps in that copy number pipeline, but they're all working towards removing the noise from the signal because exome coverage is incredibly noisy, but if we have good data about how the exome coverage varies across normal samples, then we can actually have the power to subtract that out and get variances in coverage that are only representative of copy number events. And again, we're going to use that minor allele fraction signal on the right-hand side. Those SNP calls that are maybe at 1/3, 2/3, maybe at 25%, 75%. That's good information to help support the existence of a copy number variant. So we're going to collect coverage. We're going to normalize our coverage across samples and across targets. We're going to segment all of those targets into cohesive events. And from that, we're going to call the segments. Is it actually amplified? Is it depleted? And then we can output plots like the ones you saw there with the green and orange and blue to assess just how well we did our denoising and give us a good confidence in the variance. So our newest methods are the germline CNV caller, which I didn't put up here, but is a more sophisticated model than the somatic CNV caller. And this is an overview of the structural variation pipeline that we're running now. And there's not a single tool that's out there today that uses all of the available structural variation evidence, which is strange, but we're going to fix that. So there are many tools that use depth as a signal, like our somatic copy number calling, but those tools don't use the insert size discrepancies that we would expect from copy number variants, and they don't use any split reads that we would expect from breakpoints at those structural variations. And so as we run this today, there are actually multiple algorithms that go into our pipeline, the best of the best. So there's Manta, which Illumina maintains, Delhi, which is an academic work by a very nice guy. CNMOPS is another depth caller, and then MELT for transposable elements. So once we put all of that information together, then we have a set of variants that we're reasonably confident in having pretty high sensitivity, although we know that we're still missing some of the smaller events based on long-range sequencing. But then once we have all the data from all those different callers, then the rest of the steps in the pipeline are about how do we draw a consensus across all of those. And then we use our own information to do genotyping and filtering and refinement. And then finally, there's some functional annotation that you can do on structural variants based on where they are in the exons, whether they overlap certain parts of the genes. So it got a lot of analogous steps to the SNP and indel calling pipeline, although the data is very different. So to give you a little information about our germline CNV caller, it uses a different model because germline's copy number variants are rarer and smaller. So I guess in this case, germline is actually harder. I used to have this conversation with my colleague who supervised the somatic methods, and he says, oh, germline's so easy. Here, germline is harder because in cancer samples, a lot of the time you have giant copy number variants, like entire arms being duplicated or lost, whole chromosome, whole genome duplications. But in germline, all of our cells need to be able to survive those events, so they're typically pretty small, and there's not so many of them. But we do have the nice signal that they will be, again, at these 50%, 100% allele balances that we expect from diploid humans. And then structural variation encompasses a lot of different kinds of events. Again, some of which are going to have more of one of these types of signals than the others. So for this example, the duplication on the top, the B segment was duplicated twice. It's going to have a really clear depth signal. Maybe you're lucky and you get breakpoints, maybe not. We also see inversions typically. Well, I guess that's a duplication and an inversion, which is tricky. But for just inversions, since it's the same DNA that's just been flipped, you're not going to see a depth signal. We call them balanced events because there's not DNA being added or subtracted. And so those can be a little bit harder to call. We rely more on the breakpoints. Dispersed duplications versus tandem duplications, it's usually more informative if we know where that duplicated sequence is. Is it still in the gene? Is it making an impact on the mRNA? Is it actually being translated? Or is that duplication somewhere in the non-coding region and we don't really care? Deletions, I think, speak for themselves. Translocations are kind of like what we talked about with chimeras, where you might have totally different sequence from somewhere else, just attached onto your chromosome. And then there's a surprising number of these complex events in the human genome where you might see inversions. Often there's duplications that flank either side of an inversion, which has to do with the mechanism that they start, but also part of the end repair. If there's a break in the DNA, the enzymes that we have do try and compensate for that, and they try and fill in the missing sequence. Sometimes they're right, sometimes they're wrong. So we often do see structural variants that are not perfectly described with their beautiful soft clip breakpoints, but have a little bit more going on either side. So today we'll talk about haplotype caller next, actually. And Thursday we'll talk about MUTEC2 and the somatic CNVs and ACNVs. Unfortunately, we're going to skip germline copy number for this workshop, but hopefully we should have some materials for that coming up soon. And then it's probably going to be a little while before we have structural variation documentation, but we do hope that the pipeline will be out in the next couple months. And the vast majority of these are available in our GitHub repo under GATK workflows. When we do the hands-on, we'll do most of it through our GATK workspaces in Terra, which is a nice place to go for all the best practices. In theory, you just bring your data over there, you run all the things we have, you see what pops out. So, any questions about variants in general? guess we didn't talk about transposable elements, but they're interesting. All right, so now we're going to go into details about how we do variant calling in the germline with haplotype caller. So if we look at the whole germline pipeline from end to end, we already talked about where the reads come from. We talked about how we pre-process and recalibrate those reads. And at that point, we have as good information as we're ever going to have to do our variant calling. And the output from the variant calling step with haplotype caller is going to be one of those GVCFs that has both SNPs and indels. So haplotype caller itself is effectively combined of 4 different steps. First, we identify active regions. We assemble haplotypes, because the local assembly is sort of the strength of haplotype caller, that it can resolve regions that are difficult to map, difficult to align. We're going to get the likelihoods for each of those haplotypes based on their supporting reads, and then we're going to distill those down into genotypes for each event in each sample. So the first step to identify active regions is an important one because, again, if each person has a variant, say, every 1,000 bases, there are going to be a lot of sections of the genome where it's just reference and it's not interesting and it clearly isn't causing disease. So you don't really care. And we might as well save all that compute by skipping those entirely. So what we do is initially when we evaluate any sample, we have a sliding window of bases that we look at, and we count up the pieces of evidence for some kind of variation, which can be mismatches or indels or soft clips. And then we effectively give each one of those a probability, and then we smooth over all of those to kind of generate a window of interesting things that we want to look at. We apply a threshold to that overall evidence probability function and determine which regions are active and which regions are inactive, and we can just skip when we do our expensive haplotype assembly. So the local assembly is not the same as a de novo assembly. We will still use the reference to guide our assembly. So there's kind of a limit to how big events that we can recover. But again, if we're just looking at SNPs and indels, they're smaller events and they should be well captured within that window anyway. And once we've built our graph, enumerating all the possible haplotypes based on our read evidence, we'll traverse all the paths in the graph to find potential haplotypes and then translate those into variants by aligning them back to the reference to describe how each of those haplotypes is different from the reference genome. So we always use the reference as a path in the graph. That's where we start. For example, we're going to give you this truncated set of bases here just to illustrate what we're going to do with the reads. So we have a single path, which is the reference, and then we start adding paths based on the read evidence that we see. So for example, if we see a read that has a G at the 4th position, now that's a branch point in our graph. Some reads will follow the path that has the T on top. Some reads will follow the path that has the G on the bottom. And we keep adding reads to that, and we'll discover more events, but for our little window that we're examining in our toy example, we're just going to count how many reads followed each of those paths. And so we had 7 reads in this region. 3 of them had mismatches, so 4 of them took the reference path, 3 of them took the other path. That's good support. If we have paths where only 1 read supports that haplotype, maybe 2 reads I think by default is fine. But if there's a path that's only from 1 read, that's a little suspicious. And it's also going to lead to a lot of computational complexity as we build more and more paths in the graph. So that's not enough evidence. We're going to cut that off, filter it out. So one of the beautiful things about local alignment is that it can resolve these nasty indels that happen in repetitive regions, because there's PCR error, there's alignment error, and so on and so forth. So if you look at the BWA aligned BAM for this particular region in IGV, then you'll see some events of different sizes. This one is my personal favorite, where there's a 3-base deletion, but then 1 base that's okay, but then another 7-base deletion, which is, seems really unlikely that it's 2 separate events that happen right next to each other. And so as we build the graph, it doesn't matter what the reference is. So it doesn't matter if we want to split it into a 3-base deletion and a 7-base deletion, if we want to turn it into a big deletion and SNP, or what we want to do, because it just becomes a single haplotype. And we can add in the evidence from the other reads that support that haplotype, and then align it back to the reference. And so now with a smaller set of haplotypes and restricting each read to support one of those, we can have a more concise representation of all the variation that we see in our sample. Because in humans, we only expect to see 2 haplotypes. And so we really shouldn't be seeing all these different flavors of alignments of the reads. So after we do our local reassembly for this sample, there's a 12-base pair deletion and a 10-base pair deletion. And most of the reads support one or the other pretty strongly. When we do our hands-on this afternoon, we'll talk a little bit more about what this post-align haplotype caller view is in that BAM with the artificial haplotypes. Because we can output the haplotypes that were assembled, and we can output the reads as they were realigned, and color code them based on which haplotype they support. So unfortunately, the way that BWA works when there are 2 equivalent representations is it effectively flips a coin. And so when you see a big pileup like this, some of them will be represented one way, some of them will be represented the other way. It's not particularly helpful, but they represent the same haplotype. So as we reassemble these things back together, we'll get a single haplotype. We'll realign all those reads back to that haplotype with a more consistent alignment function. And then we'll be able to transform all of those reads into one representation or the other. And which is nice, because then we'll have a clearly homozygous variant, whereas this looks like some sort of weird het SNP with insertions at different positions. Given that we look at windows of reads and longer haplotypes, from the haplotype caller, we also get phasing information based on these reads. Because in a lot of contexts, it's really important whether a variant is on the same haplotype or on a separate haplotype. For example, somehow it never makes it into the slides, but there are scenarios where you can have adjacent SNPs, and on different haplotypes, they'll turn each haplotype into a missense variant, which maybe is not great, but possibly not so deleterious. But if those SNPs are on the same haplotype, you have one reference copy with a normal protein, and then you have one loss of function, because it'll turn into a stop codon, which is going to truncate your mRNA, likely lead to nonsense-mediated decay, and have one non-functional copy, whereas we expect the dosage of 2 copies to be functional. So we can only interpret those variants properly if we know whether they go together or whether they're on separate haplotypes. And those are going to be output with these format fields, the PGT and PID. So we'll give our phase genotypes, whether if they're together, they each get 0, 1. And if they're in trans, then one of them will be 1 pipe 0 with respect to the first variant. And we'll also give this phasing identifier, because we can potentially have larger groups of variants that are all phased together. And so that represents the information that those are all related. So now that we have our haplotypes, we want to give a likelihood to each of those. So now we're looking at a bigger section of the graph, and now we have 2 SNPs. So if we enumerate all the different haplotypes in this graph, how many haplotypes are there? Yeah. So if we don't give you any information about which reads followed which paths, then you could potentially have 4 haplotypes. You can have one that chooses both reference bases. You can have one with an alternate base and then a reference, do the other crossways, and one haplotype where they're both variant. And so those are our 4 potential haplotypes, and we're going to take our reads and align them against those haplotypes to decide which ones they best support. And 4 haplotypes, not what we expect from our diploid humans. So we expect that as we align the reads back to those haplotypes, there'll be 2 that have very strong support, and then 2 that not so great. So for example, if we look at our reads, the first one has a C and a T at the variant positions, so it's going to support the path that kind of takes this diagonal. The other has a C and a G. It's going to support the path with both alternate alleles. And then this one is kind of a red herring because it has the 2 reference bases, but it also has a mismatch in the middle. And so its support for the reference haplotype is not going to be so high, not going to be as high as it could because it does have that mismatch. And the numbers in the chart are kind of toy numbers, but they're meant to illustrate how strongly the reads represent the haplotypes. So once we have our reads versus haplotypes, because we output all these variants in a VCF file with different lines at different positions, we need to break those haplotypes up into events to be able to output genotypes for them. So if we look at our graph, we really just have 2 events, each of which is a SNP. And so we will genotype our sample against each of those events. So for example, if we look at the first event, which is an A to C SNP, that is supported by 2 different haplotypes. The one that has both reference and the one that starts with a reference and then has an alternate. So we look at the read likelihoods for both of those haplotypes. I don't remember this. Haplotypes 1 and 2. And we take the better probability between the 2 of those. Let me see. Okay, this is the best support that C SNP is going to have based on this read. And we do that for all of our reads at all of our positions for all the different alleles. And then once we have those likelihoods for each allele, then we can do our genotyping. So what we want to do now is based on the likelihoods for the alleles. We know that they're going to be 2 alleles. We expect there to be 2 alleles. That's our really strong diploid assumption. But we need to figure out what they are, given that each read is only going to support a single allele by itself. So we do a little bit of math. We use the numbers in the table to plug in for the likelihood of seeing a particular read, given that haplotype. We evaluate it for each genotype, which in our diploid case is going to be combinations of 2 haplotypes, reference, 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 alt, alt, alt. We normalize those likelihoods so that they're easier to work with. And then we could potentially apply a prior, although we're going to do that in the calculate genotype posterior step in our genotype refinement pipeline. But looking at all the probabilities for those different genotype combinations, 0, 0, 0, 1, 1, 1, then we can actually make our genotype call and determine the quality. So, for example, if we go through that process for that SNP that we've been looking at the probability of the genotype given the reads, we see not a great probability for homozygous reference, looking a little bit better for heterozygous, very unlikely to be homozygous variant. And then we take those likelihoods and we Phred scale them, because again, like these small confidences are hard to work with in probability space, so we convert them to log space so that the math is a little bit easier. And then to get our genotype quality, we take the second most likely genotype. So what's the probability that this genotype was wrong? It's the probability that the second best genotype is actually right. So as we see in Phred space, homozygous reference is still the next best. It's going to give us a GQ10 for these particular calculations, which is not great. But then again, we didn't have a lot of reads, so maybe that's not such a surprise. So we'll output this in the VCF with the genotype call, the genotype quality, and the likelihoods for all the alleles. So if you're just looking at a single sample in the clinical context, or maybe you only have a single sample so far, then you're pretty much done. You have a VCF, and then you move on to filtering. But if you want to do joint analysis across multiple samples in the same cohort, then you're going to want to run haplotype caller in GVCF mode. So we talked a little bit about GVCFs in the previous presentation. That's going to keep track of the reference information for each of our samples so that we can compare them together later on. And we'll do that comparison in the genotype GVCFs tool, where we recalculate the genotypes for each sample, given a model that can take into account all of the data at once. So the command to run haplotype caller for classic mode, where you output a final genotype VCF, looks like what we have on the top. And then if you want to output a GVCF, because you're going to do analysis with more samples later on, then you add on this emit reference confidence GVCF argument, and we recommend that you name your file with a G in it so that you remember that it's a GVCF and it's an intermediate and it's not ready for analysis. So we talked about where we start with our reads. We talked about what we do to them with our local assembly, building the haplotypes, looking at reads with respect to those haplotypes, doing our single sample genotyping. And at this point, we have a GVCF with both SNP and indel calls. And next, we're going to do our joint calling across multiple samples in our big cohort. We're going to combine the GVCFs together and do genotyping, which is what John will talk about after lunch. Right? Yes. Any questions? Yeah.
To be quite honest, I'm a bit lost, so I'm not sure if I'm the only one. When you were doing the normalization of the numbers, I'm just not entirely sure where that comes from. It's just a log?
You mean this part here? Yeah, so each of those, well, they're likelihoods.
Okay, so it's
So,
probability.
yeah, so like in the Bayesian framework, the likelihoods aren't required to add up to 1 because each of them is with respect to the unknown parameter that you're trying to discover. But that doesn't really matter when we do the Fred transform. So we'll take each of those likelihoods for each genotype and plug them into the Fred scale equation. Better? I think this part is the hard part, honestly.
So you're trying to determine which haplotypes are the true ones.
Mm-hmm.
And its Fred score is trying to tell you if there is an artifact or something that it's not actually.
So we look at all of the evidence for all of the reads. Like each read can only support 1 haplotype, right? And so each read gets its own likelihood with respect to each of those alleles. And so effectively what you're going to have is at a heterozygous site, you're going to have a pile of reads, and ideally half of them will have a high likelihood for the reference and a low likelihood for the alternate, and then the other half will have the reverse, where they have a high likelihood for the alternate and a low likelihood for the reference. And so then, as you go through your genotyping, what you're going to do is you're going to start with the homozygous reference genotype, which is 2 copies of the reference. And then you look in your table and you say, okay, for each of these reads, how much does it support the reference? And so what you'll end up with is, since haplotype 1 and haplotype 2 are the same in that case, for the homozygous reference genotype, this just collapses into the single likelihood of that read for the reference haplotype. And again, like you have a nice pile of heterozygous reads, So you'll get a whole bunch of evidence from half your reads and pretty much no evidence from the other half. And then you're going to go and look at your heterozygous genotype. And so now we're going to evaluate two different haplotypes because the genotype is composed of the reference and the alternate. And we plug in the numbers from that table. For example, if we work through the numbers just here in the table, we're going to look at read 1 with respect to the reference haplotype, which is that 60. These are actually kind of funny numbers, but. And then we're going to look at the likelihood of read 1 versus the alternate A, and that's a good likelihood. So that read is very strongly supporting the A. Typically, you see the support for the other one being vanishingly small, in which case you add up all of those values, and each of them should have a good probability for the allele that it does support and a bad probability for the one that it doesn't, and then we divide those by 2. So, yeah. And then we'll go through that same exercise for homozygous variant, where we use just the one haplotype and just the likelihoods in the right-hand column supporting the A allele. And so then those, where we've added up all the likelihoods for each read, those are the numbers that go into the top of this table.
Okay, so you add up the information on all the reads.
Mm-hmm.
You calculate the table based on each read, but then you add up all the.
Yeah.
Okay.
Yeah, I mean, it's being able to calculate that table based on the whole read. That's one of the powerful things about haplotype caller. Because in like the classic SAM tools pileup genotyper, you're just looking at that one base. You don't know. But if, for example, this is sort of a toy example with the 4 haplotypes. In practice, if the SNPs are that close together, then you'll only get 2 haplotypes because they're almost always on the same read and they always support the same thing. So in reality, what's going to happen is you evaluate each read against those 2 haplotypes, and you'll either have reads that are reference-reference, or you'll have reads that are alternate-alternate. But then if you see a read that has an error, and it is reference-alternate, that second variant is going to inform the likelihood of the first position, because we look at the whole read against the whole haplotype. But for pileup, you'd say, oh, that looks perfectly fine. Like, you know, that it doesn't matter. But the fact that you have an error on an adjacent base that we also have information for gets to inform the other position in this local assembly scenario.
So it's basically a frequency thing. How frequent it is for you to get one haplotype versus the other one will tell you which one is the more real one versus which one's the frequency.
Yeah. And then, I mean, the fact that we need to break up the haplotype into alleles based on position is a convenience that we have so that we can output each variant at each position. But that's not to say that there wasn't more information that came from other parts of the read that informed that position.
But you would eventually need a read, to be able to cover the phase, you would eventually need a read that contains both variants, right?
Yeah, I mean, if they're this close together, the vast majority of your reads should, right? You have 100 base pair reads, 150 base pair reads. There's only 3 bases between those. Yeah. Yeah, and there will be some that stop after the A or start before the T, and they won't be informative for the other position, so their likelihoods will be the same for those 2 different haplotypes. Doesn't provide any evidence one way or another. Yeah, I mean, the big takeaways are, yeah, that we can do this local assembly to use all the information from the reads to try and get a smaller number of longer consensus sequences. And the fact that we do this likelihood evaluation for the whole read against the whole haplotype so that we can take information for the read and use it to inform all the bases that the read covers. And yeah, and we get phasing, and in messy regions, it does a better job representing certain types of events. Yeah, I mean, in the old scenario of very, very short reads in the early days of next generation sequencing, it was a lot more powerful. But nowadays, with these long reads, BWA does a pretty good job. There are some regions that do still get realigned, but that's, so it's, yeah, local assembly is still an improvement, but it was more necessary back when we had less high quality data. Although recently we've been working on being able to put information into the graph about which paths each read supported when there are actually cycles in the graph. So if you think about a sequence that's repetitive, you know, like these TAG repeats that you see all over the genome, then if you see a read end with a whole bunch of TAGs, maybe that's not the best example, but maybe like a longer sequence that's repetitive. It can be hard to construct from those reads how many of those repeat units were in the sample's genome. But if we add information to the graph based on which path each read followed, then we can kind of link information across multiple reads. So even though the reads may not span the full repeat, we can still recover how many copies there were. So it's just to say that there are some difficulties in the graph building, which we're addressing in more recent versions of the code. Other questions? Lunchtime?
individual sample basis, and she touched a little bit upon the joint calling workflow. So now that you've heard a little bit about haplotype caller, let's dive into how joint variant calling for germline variants actually works. So in our regular pipeline, you can see we right now are in this area where we've got data coming out of haplotype caller in GVCF mode. That'll give us some individual variants and some reference sites. We then have to consolidate them somehow and joint call them so that we can have the power of looking at all of these germline variants together to see what's real and what's a rare variant, or what's an artifact and what is a false positive. So why do we want to do this? Well, if you only have a single genome in isolation, as Laura said earlier, it will tell you how it differs from the reference genome if you run through the variant discovery pipeline. But if the reference is significantly different from the population in which that sample came from, then you're going to end up with a whole bunch of variants that aren't really variants. They're variants with respect to that particular reference, but in that individual sample, they might be completely normal variation that you would expect. Similarly, if you have extremely rare variants in a population, if you don't look at many instances of genomes from that population, it will look like an artifact or a false positive, and it'll get filtered out. So I'm going to walk through a couple of examples where we can see this sort of thing happening. So up on top, you can see that we've got this first sample. And if we look at sample one alone, well, it's not quite 50-50. There isn't a lot of coverage, so it's sort of hard to tell. There's only 2 Gs and 4 As. We're probably not going to call that. If we look at the bottom sample and the top sample together, though, and a whole bunch of samples in between, if we see enough of this particular type of variation at the site, then we could be convinced that it's a rare variant in this population that is significant, or could be significant. And we have more confidence then to call this a true variant and not to flag it as a false positive. There are other things that joint calling will help us do. So as you can see, we have another sample here. It looks pretty similar, where the orange and the blue are the read strand direction. These A alleles all occur on the forward strand, but a bunch of them also occur on the reverse strand. So it's not quite 50-50, but it's close enough. That looks reasonable. The alternate allele, this C, is only occurring on the reverse strand direction. And clearly this allele is biased for the reverse strands where we only see it there. And so if we only saw this, and there is this really high bias towards the reverse strand, do we call this variant or not? It's really hard to say in isolation. There are cases where you do see some kinds of biases in variants, but with only one sample, we just don't have enough information to say with certainty that this is real. So we can go through this sort of high-level workflow for how we do the allele bias processing. And so we've got a whole bunch of samples, and for each sample, we take a look first at this allele bias. And so since assuming humans and a diploid organism, you would expect 50-50 for each of the alleles if there was something there. But sometimes you wouldn't see that, and if you've done that for all of these samples and you still see this bias, then there could be something wrong and you probably want to filter it out. If this first allele bias filtering passes, you don't see any bias there, then we can take a look at the strand bias that we just looked at as well. And if we see that one allele only ever occurs in one strand, whether in this case it's the reverse strand or in the forward direction, we would filter that out as well. Whereas if it was a more likely variant, we would see a good breakdown where it's about half in one strand and half in the other. And by calling all of these samples together, we have enough information to see for all the samples whether we do see these things in both strands or not, and whether we have a good allele bias, no allele bias, a good allele frequency across the different alleles that we have. And again, without having many samples, it becomes very, very hard to tell if these are real or if they're artifacts or if they're something else. So let's take a look at some distributions for the kinds of things we would expect. So if we have a whole bunch of variants and we do this joint calling, oh yes?
Why is strand bias happen?
Why is strand bias?
Happen, why?
Why does it happen? Laura?
A classic example is like after a homopolymer. So, you know, a homopolymer. So like if there's an A in the reference, if you read into it from the left side, everything is fine. If you read into it from the right side, you'll see all those A's first, and then the chemistry on in the flow cell, like sometimes the A bases won't dissociate anymore after there's so many of them. And so when you have the observations of that same position after going through the homopolymer, it's more likely to be an error.
We can take a look at these annotations that we get out of this joint calling process, because we don't throw any data away, we just add annotations for whether we think the variant is of high confidence or low confidence. And we see some on the left here idealized distributions. So in a perfect case, these are the kinds of things we'd see. Centered around 0 are the real variants. And you see this purple peak over here that is off-center to the left. That would represent the false positives, or the variants that we've discovered that shouldn't actually be contained in our call set. And it's also important to note that if we have common variants, it'll be more peaky. It'll be much taller as far as the height of the distribution goes, and that allows us to filter a little bit more easily. And as the rarity of the variant increases, you get smaller and smaller peaks, and it spreads out more, where eventually you get singleton variants that are a very wide distribution across the bottom. But I should say that all three of these real variant distributions are still centered around 0, so that gives us some good confidence that we're looking at the right thing. Now in the real world, what we can do is we can look at our very large nomad collection of common variants. And we can look at another sample, and what you'll find is, because we've run all of these nomad samples together, we have a very, very strong signal around 0 and a little bit of another signal in there. It's a little bimodal, but with all of those variants called together, it becomes a lot easier to see that these are, in fact, real in that big population from which nomad was created. From a singleton sample standpoint, it's not as clear. You have a much wider distribution, and it becomes difficult to really know what is real and what isn't. And so we have, one of the tools that we run on it is this read position rank sum tool that generates statistics like this to help score the variants and then downstream to filter them out. So how much does joint calling actually help is a common question. And here's a graph of the sensitivity of variant discovery as we increase our sample count in our cohort. And it maxes out at around 120,000 samples or so. But this is a log scale, so at the very beginning we have huge increases here in the sensitivity of variant discovery. And it saturates at about 600 or so. One other thing to note, because you're probably wondering, the sample that this was run on was a very common sample, NA12878, and a lot of the common indels had already been pulled out before we ran the analysis. So we didn't capture those, and so the sensitivity is lower. So we know that it works to give you more power to find these events in a population, but what if you have important singleton mutations or variants and very rare variants? Do you have any loss of sensitivity when it comes to variants that occur very infrequently? And no, not really, as it turns out. You'll see that the singleton true positive count remains pretty steady for both indels and SNPs as we increase sample size. So even though we're adding all of these samples together and calling them as one joint cohort, we don't lose any power to find the really rare variants and singleton variants that might be in each of our samples. And that becomes really important when we want to think about how a particular sample has a genotype or another phenomenon for the clinic, for doctors.
When you're talking about cohorts, are you talking about cohorts of patients or cohorts? I don't get that.
A cohort is just the collection of samples that you run at the same time.
Yeah, but there are different patients that.
Yes.
Okay.
Different patients from the same background that,
Same background.
or from different backgrounds, but it sort of depends on what you're trying to do. But typically, like, if you want to look at an individual within the context of their genetic population, you would sequence not only that individual, but a bunch of people from the same kind of genetics, and that group together would be the cohort.
So what you're telling here is that even if you increase the size of the cohort, you add more individuals, then you're still able to accurately identify the low frequency variants.
Yes, exactly. So we also have better power to look at interesting sites around which we already have some baseline information. What I mean by that is we can look at these two sites where there are known variations that are implicated in Alzheimer's disease. And for both of these samples, if we were to process them individually, neither of them would have a variant called at that position, but it's for very different reasons and very important reasons. For the top sample, there are no SNPs in either of these sites. Or to be more specific, these variants do not occur at either of these sites in the first sample. We have plenty of data. The pileup is very large. We have tons of coverage. But these SNPs don't occur, and so, of course, we're not going to call them there, and we're not going to output that as a result. But for sample B, we also don't output anything, but it's because we don't have enough data in the case of this locus. And in this locus, something went wrong with the sequencing, and we don't have any data at all. If we process everything together, then we can have enough information, maybe, in this case, to see what's going on. Certainly not for this particular sample here, but we still get a lot better power by putting them together, if that makes sense. So it's a two-step process, and Laura talked a lot about haplotype caller already. And that's step one. And we run it in this GVCF mode so that we can get a genomic VCF file that accounts for the entire genome and not just the variants that we're interested in. Once we have that for each sample, we put all of those files together into, or we'll get there, but we consolidate all the files, and then we run the joint genotyper, this genotype GVCFs tool on all of them. And a joint calls across all of those GVCF files that we've created from each sample. So to jump back into what a GVCF file looks like, it's a type of VCF file, but it, again, accounts for the whole genome. And what you have for non-variant sites is they're in blue in this, and you have this non-ref allele and no other alternate alleles. And what that means is it's a reference site, and we can assume that that should be the same as whatever reference we're running it against. Now these sites, typically when we run it so that any adjacent reference sites get squished together. Otherwise the file would be very big, and it would be sort of redundant to say across every individual base it'll be a reference site or a variant. There may be cases where you want to do that, so it's possible, but typically for these non-ref sites, or sorry, for these reference sites, if they are adjacent and have similar properties, then they get put together. Now, when we have a variant, we, of course, include the variant allele, but we also want to track how likely it is that it is, in fact, a reference instead of the variant that we've discovered. So we add the same symbolic non-ref allele and some additional information about that non-ref allele in the likelihoods here at the back. You'll notice that, like I said, this accounts for more than one position, and this is on chromosome 20, what is it, 10,204,000 to position 210. That's all the same non-ref block, which we would expect, ref block, non-ref. Sorry for the switching of terminology, but this is all the same reference information, so we would expect it to be the reference there. And similarly, after this variant, from positions 212 to 216, it's also reference data. And as I said, a GVCF file is just a I don't want to call it a fancy VCF file, but it's a type of VCF file that just has additional information. And if you run haplotype caller in the ERC GVCF mode, then you get all the normal header information with a couple of extra fields, this GVCF block being one of them. And then you have the records themselves, and you have one record for all of these blocks of reference, and interspersed between them will be the variants that you expect, or that you have called. If you run it with the full base pair resolution, then what you get is for every single site in the genome, you have reference, variant, reference, 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 even though these three sites are adjacent. So the file will be much bigger. And so typically we run it in this configuration, and that's what we recommend. And you could do this ultimately with as many samples as you like, and what you get out is a multi-sample VCF file. So it has all of the information squashed down into a single file with all of the variants that you, all of the samples that you had in this cohort that you wanted to call together. Instead of having these reference sites, after you've done the genotype GVCF step, you end up with a proper VCF file that can be used for analysis with the per sample information appended at the end of each line. So in this case, it was joint called with 3 samples, NA12877, 12878, and 12882. And so at the very end of the line, you'll see this additional information with the genotype and some likelihoods and some other things for each of those samples. I don't know how many of you are familiar with GATK3 and GATK4, but the way we did this with GATK3 didn't scale very well. And we found that as we wanted to joint call more and more samples, it was becoming very difficult, because what we would do is we had this tool called combined GVCFs that did a hierarchical merge on the files themselves, and the most we could really get out of it was 200 samples, because it would just take too long. With GATK4, we have this GenomicsDB import tool that creates a GenomicsDB file, and GenomicsDB is it's a database or a file format that works to store data and has a backend to it that basically just runs these things much, much faster. And so this is able to scale up much, much higher than the 200 samples that we had. And what are the different ways to call it? Well, with GATK3, this combined GVCFs step, it's pretty much exactly what you would expect. You give it a reference, you give it the 2 samples or as many samples as you want, and then you name the output file whatever you'd like it to name. With GenomicsDB import, it's pretty similar. You can specify intervals though, which is nice. And you give it an output path that will create your GenomicsDB data set. It's equivalent to the output file, but now it's in a different format that allows it to be gotten at and manipulated faster. So you can use genotype GVCFs a little bit differently depending on what you have for your input data. But one of the key takeaways of this is you need to have all of your sample data together when you want to process it. You can't create a VCF file and then say, oh, I want to add one more sample to this output VCF. You need to combine everything together again and then reprocess it. And so, again, if you wanted to run the old way with genotype GVCFs or the new way, the interface is very similar, but what you give it is the reference and then either your GVCF variant file and the output, or the only thing that changes if you're running with GenomicsDB is you give it a GenomicsDB path, which corresponds to this path that you created earlier. But genotype GVCFs will work either with the GVCF file or the GenomicsDB file. But you have to give it only a single input here now, unlike the GATK3 version. I think for most of you, that's not going to be an issue. So how well does it work? Well, with the GATK3 workflow for this, we had to run on a subset of our samples. We had 3,000 whole genomes, and it took about 6 weeks to process. With Nomad, for Nomad V2, we had about 60,000 samples, whole genomes, not exomes, and it took about 3 weeks, which is 20 times more data in half as much time, which is great. So if you're doing large joint calling experiments, genotype GVCFs with GenomicsDB is the way to go. So the next things that we need to do are, now that we've got our good variants, we need to take them and filter them, refine them, annotate them, and then start doing some analysis and figure out whether they're good or not and whether we should keep using them. Yeah. Any questions? Okay.
Testing, testing. Okay, great. All right. So, moving right along this afternoon, we've just gone through both the pre-processing steps to GATK, and we just finished the center column in our big pipeline graphic, which was the variant discovery. So in the grand scheme of GATK, we've just called variants, and as John mentioned, we're going to do filtering after that. However, I want to get you guys hands-on. So I got a couple other side things to teach you, and then we're going to get hands-on on the computer. And tomorrow morning, we're actually going to be getting more into that filtering aspect of the pipeline. So first, I just wanted to remind you all, my name is Kate Noblet. I work for the Broad Institute, and I primarily run workshops like these. I've been sitting in the back most of the morning, so just wanted to let you know I'm still here. One question I wanted to ask you before I even get into this pipelining thing, how many of you have used pipelines before? Show of hands. Yeah. So I'm seeing about half. How many of you have used pipelines for your research? Yeah? What type of pipeline did you use? What was it doing?
For example, starting from Galaxy, like
Mhm.
Galaxy to do the null assembly.
Yeah.
What else? Filtering, variants, identification species.
Great.
things like that.
Yeah. Who else?
Quality control as well.
Who else had a pipeline they've run? Oh, come on. Mhm.
Variants,
Yes.
some somatic variants also to GATK.
With GATK, did you use a particular platform, or what kind of pipeline were you using?
I use the version 2 of GATK.
Mm-hmm. Okay. Anybody else want to share what pipeline they've used? It's not a trick question, I'm just curious. All right. Okay. So this talk and a little bit of the next one are going to go over information about pipelining. We're going to go a bit into the technical background so that when we do some pipelines later, you're going to understand what that means. And when we're done with that, we're going to get hands-on with that variant discovery, like I mentioned. So as an outline for this talk, we're going to go over three things. The first one is the workflow description language, which is pronounced WDL as its acronym. The second one is our execution engine called Cromwell. And the third one is a portal called Terra, and that's going to be our cloud platform we use in the end. You guys have all seen these logos in our stickers, so now you know what they mean. As I've been referencing before, the GATK pipeline itself is a pipeline, and it's a really great candidate for it. We often use these scripted workflows to automate processing. It's great to automate repetitive tasks, so you don't have to waste your time typing the same command over and over again. It also increases auditability and reproducibility. So if you're telling somebody else to run your pipeline, it's much more robust to hand them a script as opposed to telling them what to write for their script. Because it'll reduce human error. They might make typos, you may make typos. And basically just reduce time spent reinventing the wheel. As Laura mentioned at the beginning of the day, we used to just provide descriptions of what our pipelines were. And so it was pretty much, you got this picture and a long article about what you should be doing, but it wasn't anything you could just take and run. So having that thing that you can take and run with is really great. So for pipelines, we needed to find a solution that could do what we needed. We found this comic by XKCD, and I think it's really relevant here. I think most of you have probably used different pipelining options. You said Galaxy. I know at the Broad we've used a variety of different things. We've used Q, we've used Firehose, was a way long ago predecessor to Terra. Picard pipelines. Are there any other pipelining solutions out there you guys have used? Curious? No? Oh. Yeah. So there's a lot of So there's a lot of pipelining solutions out there. And we looked into a lot of the existing pipelining solutions and didn't quite find what we needed. So we decided to create another. This solution is built of 3 parts. It's got a scripting language, which was WDL. It's got an execution engine, which is Cromwell. And then it's got that UI Terra to run through it. So first up is WDL. So this is a WDL script. We're going to go a lot more into WDL itself on Friday. So I'm just going to give you a high-level overview. But the really cool thing about WDL is that this is a real script. This is what your script could look like. It's designed to be human readable and writable. It's not going to be something that requires a programming background in order to understand. We really wanted to enable people who might come from just a bench science background to be able to run bioinformatics pipelines. So right here, a WDL is basically made up of 2 parts. You've got your task here on this side, and that task has a command in it where it's going to do something. So for GATK, we run commands in our terminal. You just plug that command right in there, and that's how you'd run it. Then the pipelining aspect is where you chain together those commands, tell it what order you want it to run it in, if you want to run things in parallel, things like that. And that's where our workflow comes in. So in this example workflow, we're calling one task, and then we're calling another task. And WDL has some neat things where it determines what tasks need to be run first in order to keep everything the most efficient way possible. So that's a little overview of GATK. Any questions on this before I move on? Great. We've been going into containers a lot this morning because we had a bit of trouble with Docker. But the reason we advocate containers so much is because they are they're essential for portability and reproducibility. If you think about it, if you hand your colleague a program and just tell them to go run it, they would have to make sure they have all the same programs required to run it, all the same versions, and if a version is slightly different, they might get different results. So that's not great for reproducibility of your research. What a container does is it can package together exactly the tools and versions that you need at that time. So for our script, I think the Docker I had you download runs GATK version 4.1.4.1, and it has the correct version of Java 8 in there and the right version of the R libraries and a bunch of other dependencies that GATK relies on. But it's built up this environment for you so that you don't need to go in and individually install all those required pieces to run GATK. It's easier that way, but you can still do it the other way. We're just going a little bit the long way around to get it working for you guys since we're having a bit of trouble with Windows. So yeah, takes the guesswork out of running pipelines. Nope, skipped a slide there. There we go. So once you have your pipeline written, which is your WDL script, you need something to execute it. And that execution engine is called Cromwell. Cromwell itself is a jar that you'll run, and we'll again do that more on day 4. And it actually can take in 2 different types of languages. So today we're talking about WDL, which, as I mentioned, is designed to be human readable and writable. But there's another big player in the field of workflow languages called CWL. So if you have pipelines in CWL, you can run them through Cromwell as well. I'm not going to go too much more into it because it's a bit more complex how CWL is written. It's definitely more designed for programming types because it gives you a lot more intricate detail into your script. But that's an option there for you. And then when Cromwell runs, it can run a script on a wide variety of backends. So these down at the bottom are all different backends. It ranges from your local computer, so just running on your laptop, just entering a script and hitting run, you can do that. And you can take the same script and go all the way up into the cloud with it and run it there. There are additional options that different backends allow you to specify, but the idea is that I could take something off the cloud and run it locally, and it shouldn't fail. There are some exceptions, so there is a little bit of things you might have to do if you're doing some tricky edge case, but in general, we want to make it as easy as possible to run. Some other ones I like to point out is it runs on AWS. A lot of people use that particular cloud backend. I talk a lot about Google today because Google is behind Terra, so I know a lot more about it. Another one that people often use are high performance or clusters. Is that what HPC stands for? High performance? High performance computing. Yeah, so if you're running on a local cluster, you could absolutely set up your own Cromwell on it and run there. If that's something, some people have data security laws about putting data in the cloud, and that's a viable option for them. Any questions about this? All right. So I talk a lot about the cloud, and I think it's important to take a step back and think about why we're going to the cloud. And one of the big reasons is that cloud makes it just so much easier to share data. If you look here on the left, you have your traditional way of sharing data, which is really bringing data to the researchers. You've got this data repository, but then each individual researcher would download the data off of it. If you think about it, the human genome has to be copied like 1 million times across the world. It's being stored in so many different places on so many different computers. I have a copy of the human genome on my computer, and I don't even really run pipelines all that much. It's wasted compute, globally. But on the other hand, if you were in the cloud and you just had one single place where it was stored and everybody could reference it there, just point there and say, there it is, use that, it saves a lot of data space that way. And actually at the Broad, I believe we host the human genome in a public bucket, so you can just access it through that. We recently moved it to that, so we're really excited about it. So in that way, you move from data copying to really true data sharing. Another really important thing about compute is that in the old way, it requires big infrastructure at each site. So if you're trying to run a big pipeline, you need to buy all the computers, you need to set up all the computers, have a room there for it, hire an IT guy to maintain them, and you end up with largely fixed compute. You only have as many computers as you bought. So if you need more compute, you have to go out and buy more computers. And additionally, there's individual security implementations. So your security is only as strong as you've made your security at that site. So you're subject to, I know audits are a big issue in the US for keeping human subject data private, and I'm pretty sure they are internationally as well. But with the cloud, you have the cloud providing the infrastructure. You have elastic compute and storage, which means it's flexible to expand or contract based on your needs at the time. And you've got this centralized security implementation. So Terra is, shoot, I used to know this. It's FISMA moderate certified. Do you guys know? FISMA, we're doing something with FISMA. Yeah, there you go. So we're certified FISMA moderate, and we're also going through some FedRAMP certification at the moment, which are big, high security things, but we're putting that effort in there, and Google's putting that effort in there so that you guys don't really have to worry about, is my data going to be hacked? Because you have all these auditable security implementations in place. It's also, Terra itself is designed in such a way to prevent you from accidentally sharing data to the wrong person. If you are actively going out of your way to break those rules and go around it, it's still possible, but it'll prevent you from accidentally sharing data with someone who shouldn't have access to that data. So, yep, you've got your data being downloaded and hosted locally, and there's all sorts of complications associated with that. Or you have the cloud, where it's got this one central place that everybody comes to access their data. And that's what we're trying to build with the cloud, and that's what Google's trying to build, and Amazon's trying to build, and all these various cloud providers are working towards. So this is a graph of a very big reason why the Broad moved to the cloud. These are genome and exome processing requests per day over several months. This is from December 2018 to March 2019, so beginning of last year, really. And you can see that it's really spiky. If we bought computers in order to handle the top levels of the spike, there's all this time where we're not using the computers to or the compute cluster to its full abilities. We're paying the overhead of keeping them and keeping them maintained, but we're not using that thing. On the inverse, if we chose to buy like enough for average of like what we usually get, you end up with like these backlogs where sometimes it just keeps increasing and keeps increasing, and you end up with so much data to process that you just have this backlog of things that we'll get to when we have more time, when things calm down. I remember when I first started at the Broad, before we had moved to the cloud, we actually ended up having to take compute away from the developers in order to give more compute to processing these genomes. So it was a real problem, and it's a big reason why we moved to the cloud. Now, that's all well and good because we're a big sequencing center, but does that really apply to you guys as like just a researcher at a smaller level? And yeah, it totally does. Think of conferences, for example. 2 weeks before a conference, what's everybody doing? Running their analysis. You could get stuck in a queue behind everybody else running their analysis, waiting for time to free up so that you can run yours. You can also, yeah, you'd just be stuck in a queue. I think that was my only example. But yeah, this is a large reason why the cloud is nice. It can expand to fit your needs at those times of high demand, and you're paying for that high demand when you need it. And then when you don't need it, your costs go way down. You don't have to pay for it. Question? No? Any questions? All right. All right. So this particular graphic shows you our setup for how we work and develop in the cloud. And it's actually both in the cloud and locally, which is interesting. So our researchers will write a script or have a script they want to test and work with, and that script can have command line tools in it like GATK or other tools that we're working with. You run this script to Cromwell, and Cromwell can launch that either on our local backend, which is a SunGrid engine cluster, and it'll run things. A cluster like this is designed to run like maybe a full-scale sample, but not necessarily like 10,000 samples. So we would run like our tests there, making sure the script works, making sure the logic of it works, making sure things are running in the right amount of time. And then when we're happy with the script itself, we can take that same script, run it through Cromwell again, and launch it to the cloud. And in the cloud, Google's Pipelines API, or POPI as you'll often hear it called, that will run it in the cloud, and it has a lot more space. And that's where we do our production level variant calling on like thousands and thousands of samples. So, yeah. So there are a couple different paths to running GATK pipelines in the cloud. You can run directly from the command line. So if you open up your terminal, you can run directly from GATK using Spark. Or you can use Cromwell directly. I'm going to show you how to do that locally, but there's also a way to set up a Cromwell server, for example, on your cluster and launch directly to that. You can use that. But the one we'll be talking about largely throughout this workshop is Terra. Terra is a web graphic user interface, basically a nice user interface for you guys to access and work with the scripts in without having to necessarily be really strong in coding, but still giving you access to APIs if you want that access to work more in detail. So what is Terra? Terra is, like it says in the title, self-service analysis for all. It's this cloud-based platform, and it's built on top of Google Cloud Platform. It's free to access, meaning we at the Broad don't make money off of this. The only things you're charged for Terra is actually Google. So if you're using Google's computers, Google is charging you for the time that you're running computations. They also charge a smaller amount for storage of your data, although there's flexible options. You can put things in cold storage if you're not using it as much, things like that. Another great thing about Terra is that you can access published data. We have a lot of data sets in Terra now. And you can access existing methods. You also have the option to upload your own data, upload your own methods, and do novel things in the cloud. You can also execute analysis on auditable pipelines. I think I show one later, but we've shown you before all the featured workspaces that we have in Terra. We have the genome variant discovery that we're talking about with GATK. We have that pipeline in the cloud. You can literally just clone the workspace, put your data in, hit run. It's that easy. So you can execute those analyses really easy. The last bit is sharing data. If you have a result in the cloud, you can share that data in the cloud. We have, you can link a workspace in your paper that you publish and know that people can go directly to your workspace, clone it, and reproduce your analysis very easily right there. Any questions on this? Okay. All right, so I like to show this graphic to show you a little bit about how Terra is built and how the different pieces connect together, sort of conceptually, before we get into the actual platform. So it's a lot to take in at first, but let's see if we can break this down. So you as a researcher, maybe you're coming in and you're writing a script yourself. Maybe you have some novel pipeline that you want to run. So if you've written a script, you can upload it to the cloud, and we call them workflows. You'll hear me talk about workflows or pipelines kind of interchangeably. They essentially mean the same thing for our purposes. So when you upload your WDL script to the cloud, you'll put it in a methods repository of some sort, and that's where the script lives. If you haven't written one, you can still access one that's already in the cloud that somebody else may have contributed. The other big entry point we often see is you have your data that you're bringing to the cloud and you want to run an analysis on it. So for that one, you would create something called a workspace, which is this big green circle here. And a workspace is essentially your sandbox in Terra, where you can upload your data. It'll go in a bucket. I'll talk about buckets more later. You can grab a configuration. So configuration is basically your workflow telling it, this is the data I want to use, this is the script I want to use, here's how they plug together. And you fit it all into something called a data model, which is how you fit things together. And then when you have your data and you have your script that you want to run, you're going to hand that over to Cromwell, which is our batch execution engine. It packages up all your workflows. Very often you're running more than one workflow at once. It'll package them up and send them off to Google, where Google's pipeline API does the rest. It runs the computations, it creates the files that you create during your pipeline, and then hands you back at the end your results, the things that you told it you were interested in finding about. Any questions about this? All right.
How much it costs processing per?
Processing? So it varies depending on how much time your pipeline takes, how big the files are, things like that. I'm going to show you a variety of workflows throughout this workshop, and we show you how much those workflows cost. It honestly surprised me how little it was. For example, we have a $5 genome pipeline which runs GATK's best practices for $5. It's a bit of a misnomer now because Google recently, like last week, changed their prices. So it's like $7 or $8 now, I think, but still pretty inexpensive for a whole genome running through the pipeline.
It's faster now.
It is faster, yeah. Yeah? Yeah. Yes. All right. Okay, so last two slides here. This one is showing you some workflows preloaded into Terra. I mentioned our featured workspaces, so these are things that you can find showcased in Terra. You have on this side a whole bunch of GATK example workflows. We have our germline best practices, we have our somatic best practices. There's one for mitochondria, like way down here, that we're gonna go over on Thursday? Yeah, Thursday. And then we also have these like other featured workspaces, which isn't very well named because there's a wide variety of use cases. So we wanna make it more understandable. But essentially these are other workspaces that people have contributed outside of GATK, which you might also find very interesting. We're gonna be tackling one tomorrow about a reproducible GWAS study, which is pretty interesting. The last thing that I haven't mentioned yet about Terra is something called notebooks. How many of you have used notebooks before? Jupyter notebooks? Okay, a few. Jupyter notebooks can be thought of, I don't know, I like to think of it as a digital lab notebook. You can have blocks of code that you run, and then you can also have blocks of text. So you can say what the code's about to do and then run the code right in line. You don't have to have like a document here and the code executing somewhere else. It's really nice to display that way. And we find it really useful actually for running tutorials with you guys because it has all the information right there. I'm going to be running some tutorials with you today, but the documentation in them on their own should be able to be run on their own. So you can take them back to your lab and have your lab mate run them as well, and they should understand things, or you could run them again if you want a refresher. All right, so that is notebooks. Any questions on this so far? All right, let me have a moment to switch over slides then. Hearing a lot of chatter. You guys sure you don't have questions? Yeah? Yeah? Yeah, so on a local backend, Cromwell is what, so when I say local backend, I mean your computer. It's what would take your text file, which is your WDL script, it translates it and parses it into your computer doing something. On the Google backend, it's a little bit different because Google has its own API and own ways that it wants to run. So what Cromwell does in that case is it packages together various workflows into a big lump of workflows because you can only launch, I think it's like 10,000 at a time. So it groups them up into groups of 10,000 max and then sends them to the pipeline's API. And it tells the API calls how to translate a WDL script into executable actions on the cloud computers. Does that make sense? Okay. Yeah, no problem. Any other questions? All right. So next up, we're going to talk a little bit more about Terra, and I'm going to dive a bit more into the details of how you use Terra. Hopefully this isn't too repetitive for you and you find it interesting. One thing I like to point out right at the start is as scientists, you're used to having a certain research process, and translating that into the cloud can often be kind of a confusing concept. So, like, one of our graphic designers made this, and they're really awesome. You can take data in, you're going to do some form of analysis in a research project, and you'll end up with a result. So what this means in the cloud or in computational land, you'll have some input of data and resources that you're doing. So for the germline variant discovery pipeline I've been discussing, our input is BAM files. You do some sort of automated processing portion, and that would be our pipeline. So running that pipeline of certain steps. And then after that pipeline's over, you want to do some sort of analysis on it. You want to really dig into the data and query it. And we're going to actually show you more about how to do that in the cloud as well. And then lastly, you want to share your materials and methods, because what's the point of research if you can't talk about it? So in this example research, they took in some exome sequencing data, and they had a WDL script for mapping and variant discovery. And then they used Jupyter notebooks for doing some prediction and variant load analysis. And then they presented their paper in bioRxiv, which I'm not sure if that's a link. Yeah, that's not a link. Never mind. There is a bioRxiv paper that did link Terra. I'm sure there are a few at this point. It's just one particular one. Nomad SV? Yeah. All right. So looking a little bit more at the inputs. This is a small breakdown of that very large graphic I showed you on the last slide deck. And I want to dive a little bit more into this bit. So your workspace is your computational sandbox. It's got your methods, it's got your data, it's got your tools all in it. Your data itself, it can be stored in a number of different places. So when you create a workspace, it comes with its own Google Storage bucket. And by default, whenever you run a pipeline, your results are returned back into that bucket. But a great thing about working in a workspace is that you can access any Google bucket that exists. So if there's a public bucket at the Broad where we're hosting a reference file for you, you can access that bucket by just typing in the URLs for it. There's also a whole bunch of data stored in our data library, which is what that top bucket there is. But essentially, you can pull in data from a variety of different sources in order to empower discovery for your variants and empower discovery for your study. All of that data is tied together in something called a data model. So the data model is what you'll see in Terra. It's essentially a table that tells Terra, this is how my data is organized. So instead of just having a list of URLs of where all your data files live, you have it neatly organized in this data model so that you can track it easily. There's another place where data is stored, which isn't mentioned in this figure, but I do want to bring up here. Notebooks themselves are kind of like spinning up your own virtual machine, and those virtual machines have their own data inside them. So when you spin up a notebook to do some interactive analysis, it has its own set of files that you could use in there. A notebook can go out and access a storage bucket. It can copy things to it. It can copy things from it. But you can't, since notebooks are like temporary things that you spin up and shut down, you can't call a file that lives in a notebook. So if you did an analysis in a notebook and you produced some results, you want to copy that out to a bucket so that you can use it elsewhere and just store it in your data bucket. So I'll remind you about that periodically because it comes up. So I mentioned the data model, and a big question that came up with Terra's predecessor, FireCloud, was about how data should be structured. So the predecessor to Terra was built with cancer in mind, and as such, it created some basic types of data that cancer often used. Nowadays, Terra can do more, and I'll talk about that in a second, but we have these these two basic types and some additional types of data. You can organize your study by participants, where you have a certain participant ID, and that participant can have many different samples taken from them. So they might have a whole genome sequence done, they might have a gene panel, you might have some metadata about like their age, their phenotype, things like that you're studying. All of that can be stored within the data table, and it would be stored in a sample data table. Additionally, we have these other data sets. So you have pair as an option, which is, again, designed for cancer to do the tumor and normal pair. And then you have sets, which are groups of any of the three other types. So if you want a group of participants that you're launching a workflow on all together, you can do that, or groups of any of the other types. The interesting thing that Terra adds is that these are not your only types. If you need a table for like something completely different from this, I think we have a dog aging project going on right now, and they need to use a very interesting structure for their data tables. So they actually just created a new data table, and you can name it whatever you want. It's like opening an Excel spreadsheet. You can just type whatever you want into the columns. It's not that simple because you don't actually have columns to type things into, but it's structured like an Excel spreadsheet, I'd say. Any questions on this? Make sense? Great. All right, so the next step after your inputs is your processing. And I've already spoken a bit about this workflow description language. So I'm going to kind of skim through this one a bit. You've got your workflow, you've got your tasks that you're called in a certain order, and you plumb them together by taking, so task B, Cromwell knows runs after task A because it depends on an output from task A. So when it's parsing through the file and deciding, oh, what commands am I going to tell your computer to run, it sees this and it knows that this has to go second. It has to go once task A finishes. pretty neat. And then another interesting thing to point out in task A over here, I mentioned the command before, but I haven't mentioned this runtime. So we've talked a lot about containerizing our workflows, and this is actually where you specify your container. We have Docker images hosted in the cloud, and we'll be using one later when we're actually working on Terra. But you can just plug in the URL for existing Dockers and use that as your runtime environment where you're going to be running your, that particular task. The really cool part is that you can adjust those runtimes for every single individual task, meaning if a certain task needs Java version 8, you can do that, and the next task can run on Java version 13. It's not a problem. Yep. All right, so again, this one dives a little bit deeper into Dockers itself. You can build an image following some Docker documentation. We also host all of our Dockers on the Google Container Registry. We used to host them on Docker Hub, but we used them too often, so they told us to stop. We were actually DDoSing their servers by referencing the Dockers so often. So when you create your container, you can make them public or private in this Google Container Repository. And you can just, this is how a Docker image is referenced. It looks just like that, which you'll recognize is the same thing you were typing into your command terminal. Running little workflows. All right, I've already gone through this. I'm sorry. I should have viewed these slides a bit closer. It seems like these have a lot of overlap. Are there any questions on this overall idea of Terra, or are we pretty much understanding? No? Okay. And I've talked about virtual machines. And here, this is an important part to show you. When you're ready to run a workflow, this is actually the screen you'll be presented with. You have the name of the workflow that you'll be running. You have a synopsis of what that particular workflow is doing. And this is a synopsis entered by whoever authored the method itself. So documentation is great, guys. And then you have two options. You can process a single workflow from files, which just means I don't want to set up my data model. I don't want to put things in my Google bucket. I know exactly the URLs I want to run on, and I want to type them in. That's great for running the first time. It's not easy to redo because you have to change the URLs every time, but it's a way that we often go for just running simple tests. The great thing is the second one, which is process multiple workflows from, and then there's a drop-down. That drop-down actually lets you select what you want from whatever table you're working with. So in this one, it's using samples. So what this means is it's going to run this workflow on every single sample that you select from this sample thing. And you just hit select data to select the samples that you want. Another option you have is to select a sample set. So if you have like a pre-grouped number of like 6,000 of them, you don't have to go in and select 6,000 every time. You can create a set for it. And then lastly is our tertiary analysis with Jupyter Notebooks. So, oh, this is one of those. My colleague really likes animations, and I can't do that. So Jupyter Notebooks is an open source web application. So we've actually put Jupyter Notebooks into Terra itself, so you're able to access the data that you have in your workspace in a very convenient and intuitive way. It's great for live code. You can put equations in there, visualizations. We're going to show you a lot of plotting stuff this week, which is interesting. And then narrative text. So I talked about documentation within the notebook as well. And then the other bit to mention with Jupyter Notebooks, we've talked about how you can plug a Docker in for your workflow, but how do you control the environment for your notebook? And that's done through this notebook runtime. And it actually looks a bit different now because you can specify a custom Docker image up here. You can say, I want to run on this Docker image that I've created, and that's easy enough to plug in. But otherwise, you have these profiles you can select, which determine like how many CPUs, what your disk size, what your memory, and all of that's going to affect the size of the machine you're requesting from Google. And the size of your machine tells it how much it's going to cost. So cost is really important. People definitely care about how much they're spending. While you're working and the notebook is actively running, this particular notebook costs 56 cents per hour to run, which means when you're actively typing in it and it's running, it's going to cost you 56 cents per hour. Notebooks auto pause after, I believe it's 30 minutes of inactivity, or you can manually pause it. And when your runtime is stopped, so this one is paused, it's currently stopped, it's costing us 1 cent an hour. And what that does is it maintains all the files you've created in there without like deleting them. It just pauses everything. And you can pick up your analysis again tomorrow and go back up to the more expensive costs of actively using the machine. So in that way you can definitely save yourself money. And then when you're done with the analysis entirely, you just delete the runtime and it'll delete your files, anything you didn't copy out to your Google bucket, but the cost will be back down to 0. Any questions about that cost part?
So if you pause it, your cost stops at that point.
If you pause it, your cost goes down significantly. It's usually 1 or 2 cents,
Okay.
depending on how much data it's like holding on to for you.
So it's flushing it down to 0. That actually stops it goes back to 0 cost.
Yes. So
like if I want to if I'm working on my notebook and I just want to I don't know, go to bed, and I don't want to keep it running.
Yeah.
So I would pausing it wouldn't make it stop. It would just lower the cost down to like, I don't know, 2 cents an hour or something.
Yeah. So pausing it is like putting your computer in sleep mode for the night. You can't run any programs while it's sleeping, but it'll cost you a lot less because Google knows that you're not actively using it.
Yeah.
But it still keeps your files on it.
Okay.
When you delete your cluster, it's like throwing your computer in the garbage. You don't have those files anymore.
Well, that is correct.
But it doesn't cost you anything anymore either.
Okay.
So before you delete your computer, you want to copy those files that are important to you off of it.
I'll put them to a bucket.
Yes.
Okay.
Yeah. Yeah, we'll show you how to do that too. Question?
Is there any way to calculate, estimate how much time it's going to
Take you to run?
take you to run so you can have an estimate of how much it's
Yeah.
going to cost you?
So for our featured workspaces where we're giving you a notebook, we can estimate that for you. We'll tell you this is going to cost you, it's usually like less than $1 because it'll take you less than an hour to work through this notebook.
It's only because you have run it before.
Yes.
And you know how long it takes. But,
Yes.
but
So
if it's the first time that you're going to run your workflow,
Yes.
then you
So-
don't know until the
So these
second.
aren't workflows, actually. Notebooks are for interactive analysis. So after you've run your workflow in the cloud, you've run through the pipeline, you come up with a VCF at the end, and now you need to do like some filtering or refinement. That exploratory phase, it's really dependent on you, how long it takes you to query the data and how long it takes you to decide what you want to do with it. That's how long it'll take. So that's why we can provide estimates for ones we've written, ones we've run through, because we know how much material we put in there. But typically it's more of an interactive, exploratory thing.
Sorry, it's one alarm.
So not everything all set up when you want to do a histograms or just
Yes.
scatterplot with Seaborn or something
Yeah,
like that.
yeah, absolutely.
You basically do that, with a notebook. You're not going to do like some heavy analysis. with that.
Yes.
That that definitely makes sense.
Yeah.
So, that you're not going to do like a 10-hour or something, just running processing, because otherwise you have something else. Other alternatives include like Google Colab. Because
Okay.
that's why Google Colab works for what you're asking.
Okay.
You got Google Colab, you need GPU or something like that. It's Google Colab.
Question?
If you need to change your runtime environment while you are working, are you able to do so, or you must create a new one?
So You will be able to edit any of these settings. When you edit them, it will slowly apply those settings and then ask you to apply them. When you do that, it does delete your old runtime and create a new one. So your files will be gone. So you'll want to copy them to a Google bucket before you do that. Yeah. All right, any more questions on notebooks and their cost? Okay, I wanted to show you this thing down here. So this is the notebook terminal. You'll recognize it that it looks very similar to your terminal. If you're comfortable using the terminal, you can use that to run commands that way. But you can also do it within the notebook itself by just creating a new cell, which honestly, I find the notebooks themselves easier to use than this terminal, but it is there as an option for you if you'd like. All right, that is all I have for slides. I'm going to real quick show you something in Terra. So I want you all to open up your computers and go to Terra and log in. If you haven't signed up, please sign up now. The very first thing that I need you to check, there might be a banner at the top of your screen here that says, click here to activate your free credits. You're going to click there and activate your free credits. If you already did so, then it might say your free credits are active, or you might have closed the banner and it might be gone. But we just need to get you on a free credits account. Laura, John, can you see my screen from the back? Text okay? Or should I zoom in? Good? Okay. So once you're on Terra, we're going to click on this View Examples button in the center, and that's going to take us to our Showcase workspaces. So these are all of our featured workspaces. If you scroll down, you see there's quite a lot of them. The one we're interested in right now is titled GATK Tutorials-Germline, which I'm just going to Command F search for it. There you go, GATK Tutorials-Germline. And you're just going to click on it, and that's going to open the workspace for you. And hover here so you see the name. If at any point you fall behind while I'm giving a tutorial, raise your hand. What do you need?
What's the step before this is?
So you're just going to go to app.terra.bio and log in. And then when you're on the main page, you'll click on View Examples.
I'm in terms of use of the free credit thingy.
Okay, so you'll need to accept the terms of use.
Okay, accept.
Yep, so click those and then hit accept. Yep. The free credits? Yes. So there should be a banner at the top of the screen. It should say click here to activate your free credits. It'll pop up and give you a terms of use to read. So you just scroll through that, click accept at the bottom. There's like 2 check boxes, and then you hit accept. Do you see that? Okay. Okay, what happens when you click that? Okay, so go back to the Terra. Yep, and then open up the hamburger menu there, and then click on your name, the little drop down next to it. I want you to go to billing. Okay, so your free credits are already active. You have one. Yep, you're good. And that's why I wouldn't send you anywhere.
Okay, thank you.
Yep. So that one's telling you to start your free trial. So you should click there and accept the terms of use. So you just have to scroll all the way down, hit review, check the boxes.
We use that platform, right?
What?
We use that platform
Yep.
tomorrow.
Yep, we're going to be using it all throughout the course. Yep. So you're all set. All right. So who else needs help activating their free trial? Anyone? Yes? You've got it? Okay. Then what we are doing next, are we all caught up or do we need help? All right, show of hands, who has their free credits trial activated? Activated and good. All right, everyone does. So I'm going to move on. Your next step is to go to the View Examples button, and it'll take you to this page. You're going to find this GATK tutorials-germline. This is the workspace we'll be using today. So if you click into it, you're presented with this screen. You'll see at the top it says Read Only. So the very first thing we need to do is go to these 3 dots here, and we're going to clone it. So when you clone, you're presented with this pop-up here, and the first part is to rename the workspace. So you'll want to name this something that's identifiable to you. Typically what I like to do is leave the base name of the workspace alone, but I'm going to add my name to the end, like that. Now, workspace names have to be unique to each user per billing project. So if you and I are on the same billing project and we both named this workspace Germline Costa Rica, then the person who hit clone second would get an error message saying, oh, it's already taken. So,
Wait, so it's per billing, not
per billing project, yes.
not per user per billing, but just per
True,
billing.
yes.
Okay.
Yeah, I guess it's per billing project. I was thinking like if I tried to name a second one, it would also fail. It's per billing project. So in the billing project section, you should have only one option. It'll start with FC credits, and then it has some randomly generated words and numbers there. This is the billing project you should use. If you don't see anything here, please raise your hand. Let us know. But once you select that, you should leave the description alone, leave the authorization domain alone, and hit Clone Workspace. So go ahead and click that button when you're ready.
So you have to select the only one that is showing there.
Yes, you have to select the only billing project that's showing there.
Oh, okay, got it.
Yep. But leave this authorization domain blank. If anybody's having trouble, feel free to raise your hand. We have some TAs that can come over and help you, or I can help you. Question?
Yeah, so I do get the billing project, but I get this weird exception error.
Okay, let me see.
There it goes.
Okay, so did you just accept this billing, or the free credits? Or is this an old one?
No, is this a new one?
New one?
Yeah,
Okay.
I created this one.
All right, so I'm going to hit cancel, and I'm just going to refresh the page real quick.
Yeah.
Sometimes it has some difficulties like connecting it.
Yeah,
So I'm just going to try cloning again, selecting this. And then, how did you rename it?
that's okay.
Then try hitting clone again.
All right. Suggested maybe they expired. The billing, the free credits? Yeah. I think it's like 3 months. Well, they expire after 30 days, but this is a new billing project. Or, no, it is like 3 months, but these are new. Like, they just signed up.
But you were at a workshop before, right?
Yeah, but 2 years ago.
But
If you've
it's
used,
the same username, right?
or did you create a new account?
No, I used my old email account.
Okay.
Yeah.
Okay, so then yours is expired, and that's why it's failing.
Okay.
What we're gonna do is add you to a different billing project real quick.
Hold up.
Yep. Can I ask you to type your email in here? Is everybody else able to successfully clone the workspace?
I just leave the authorization domain in blank.
Blank, and then just hit clone. Yep.
How many times how many time is the free access?
Okay.
So the free credits are $300, or I believe it's 3 months, whichever happens first. So if you spend $300, that's it. Mhm. Okay, so now try refreshing your page again.
Yeah.
And then when you clone your workspace, this time you'll select workshop-temp from your billing projects.
Okay.
And that one you'll use. So it's just a temporary billing project we add you to for this workshop, so you'll lose access to it after, but it'll allow you to run for today and not spend your own money.
That makes sense.
Yep.
Workshop temp.
Yep, there you go. All right, so now that you've cloned your workspace, you should see this view here. The first thing we need to do is start up a notebook runtime environment. So I have one that's paused because I ran it last night. But what we're going to do is set up a new one with the following settings that I'm going to enter. And then the script is this. All right. So the options you want to select is from environment, you're choosing legacy. This is the place where you could put in your own custom Docker image as well. For compute power, we're going to select a custom profile. Basically, we want 4 CPUs. It should default to 15 gigs of memory, and then you want to increase the disk size to 100. The important part is this startup script here at the end. This is a startup script that's going to install GATK into your clusters so that you'll be able to run it. Run GATK while in your notebook. And make sure that this is just left unchecked. We'll use that in a different part of the workshop, but leave it unchecked for now. And then yours at the bottom should say create instead of replace, so you can just, yep. Is that too small? Do you guys want it bigger? I don't really have a way to give it to you guys.
No, okay.
Sorry.
I didn't see how you
Yeah.
get it.
Oh, actually, yeah, you can copy it. It's right down here. It's in this table. If you scroll down on the dashboard, you can copy it from there. Yep.
Yep, then you hit create. So this is the warning when you hit create or when you hit replace, I guess. Replacing your runtime will delete files on your disk. So we had that question earlier. It does tell you. It's nice to know. So you'll hit create, and then it should change over to creating like that. Now, this particular runtime environment that we've set up for you guys today is costing us 20 cents an hour, so pretty inexpensive in terms of runtimes. It's going to take between 5 and 10 minutes to start up. It tends to lean more towards 10 minutes when like everybody presses create at the same time. So in the meantime, let me see, are we running ahead? So we have about 15 minutes for the coffee break. Do you want to give a coffee break or do you want to just keep going? What do you think?
We can make the coffee break.
Have a coffee break? Okay. So while your notebook is spinning up, we're going to have a little coffee break. So unfortunately, we're running a little bit behind in terms of presentations. So we only have 15 minutes so that we can still get you guys out by 4. So feel free to take a walk, go to the bathroom, get water, things like that. But we're going to meet back at 2:50. We'll meet back at 2:50. Yes. Now you can take them. So that is the GATK OWL. His name is Mo, and it stands for the GATK Nightly Builds. Do you know Geraldine? Okay, she is the original person who created the GATK forums and ran support for a bunch of years. So she is affectionately known as the benevolent dictator for life of GATK. And she actually created that logo. So she was asked to make a logo for the GATK Nightly Build, sort of as a joke, and then she did it, and now we give it out as stickers.
Oh, cool.
Yep.
We gotta put it on our Dogger Nightly page somehow.
Yeah.
Because we don't have like actual Nightly builds anymore.
No, we don't.
Just the Dogger Nightly.
But it's nice to have a mascot. I mean, we have mascots for all the other stuff.
So yeah, exactly.
Yep.
It's nice to have one.
Yep.
Can we get some?
Yeah, grab as many as you want.
We brought plenty.
Yes. Do you know if we have this room for all 4 days, or are there other people coming in?
No, I'm pretty sure we are.
Just all? Because I'm just going to leave those there so I don't have to carry them.
You should.
Yeah.
Yeah. Yeah, it will be about, it doesn't make sense to just carry them around. I mean, it's not gonna be one of those scenarios. I would have left those stickers there. It would have been for those pesky kids.
I've never had a problem with people stealing them, but I do like forget them for more. I figure anybody who wanted one grabbed one. But then every time on the last day, people are like, "Where are those stickers?" It's fun.
They will take them.
Yeah. Oh, good.
If you leave them, they will come to them. God, just stop. Just
Yeah.
such a
Copy?
I'm just gonna grab water because I've been talking a lot.
Yes. It's mandatory. At 3:30, it's mandatory. Between 3 and 4, it's eventually it will become a law. There's unspoken things, but it is a law. Like this country stops every day between 3 and 4, and everyone, it's like, where's everyone? Just
taking coffee.
yeah, drinking coffee. Probably with some sort of like underdeveloped pastry because it's not very good. Usually like just bread. Yeah. It does explain the fudge. But we do love coffee and bread.
I had to buy my own coffee in Montreal. That's how bad the hospitality was. Yeah.
What?
Unheard of.
In Canada?
Yeah.
Canada?
Yeah, but the French-speaking
Montreal,
Canadians are the
yeah.
worst of the Canadians.
Well, I mean, that is the French part,
Yeah.
isn't it?
Yeah. Stingy.
I remember Belgium had this really great espresso machine in the room.
Oh, wow.
So anytime you weren't presenting, you were just back at the espresso machine.
Was it super loud?
No, it wasn't.
Oh.
Yeah, it was nice.
Was it one of the single cup like Nespressos? Or
Yeah,
was
so it did espressos and then it did mochas, which I really liked.
Nice.
I'm a chocolate person.
The hot chocolate out of the machines at work is very,
They're pretty good,
very
yeah.
It is, yeah. It's like Swiss Miss or something, but
Yeah.
it's still really good.
So Rodrigo actually, he was, we had a coffee place on campus, or it's kind of like stalls around, they sell coffee. It was really good. For some reason, they got kicked out of campus. I don't know. I mean, that's just insane. But Rodrigo's concocting alongside all the professors in the microbiology department to buy this tiny espresso machine. And it's really good. And he's trying to get me into putting in some money. And I feel kind of bamboozled because I work in the computer sciences department. So to actually get a cup of coffee, I had to walk like almost a mile. So he kind of describes it as the following way. It's the best darn coffee ever. It's totally worth walking a mile. I was like, yeah, but is it really? Is it really? I mean, go for the coffee, stay for the conversation. It's like, yeah, I mean, the way you sell it, it's really worth it. Every time I go into his office, I gotta tell you, it ends up being fantastic because it comes out into a paper, it transforms into a paper, so I'm inclined to actually participate in buying in the purchase of that coffee machine, because the amount of papers that could come out of that outweighs the coffee machine. But
Depending on your contribution, then you will get the authorship that they order.
exactly.
Yeah. Someone explained that to me when I was a grad student, like what all those middle authors are for. And they're like,
Yeah.
well, I was somebody who was just like brainstorming in the beginning, and now I'm one of those middle authors. It's very exciting.
Oh yeah. Coffee, Rodrigo?
Yes, go.
I've done Thule's RNA-seq paper because I looked at a couple of variants for her. I'm like, rock on. It's like GTEx something.
I'm gonna just check people's computers and make sure they hit run.
And what are you gonna do if they didn't? Are you gonna
Hit-
touch somebody else's
hit-
computer?
Hit run for
You're gonna
them.
touch somebody else's computer?
If they left it unlocked, I can click a button. But nobody else seems to have left it unlocked. All right, that's running. All right, that's running, great. Now the mic is on for the camera. This one? Yep, just refresh the page. It's because we've been sitting on the page for too long. You can refresh the page just fine. Yeah, it's stopping now because we took too long a break. So runtime environments pause when there's been no activity on them. So because it started up so quickly and then we went and took a half-hour break, it's stopping now. So all we're going to do is see how the unpausing portion works. So we're going to let it stop and then restart it when it finishes. While it's stopping, though, I have an opportunity to show you a little bit about the workspace we're in. So you don't need to do this, but I'm just going to open up a new tab to look at this. And what we're going to do is show you here that the workspace is built up of multiple different tabs, and you might recognize some of these names from the slides that I was going through. This is what it looks like in practice. The dashboard is the first page you always land on in a workspace, and it's very often full of documentation about what is included in the workspace. In particular, our featured workspaces, which this is one of them, has a lot of documentation about what we're planning on doing today during this workshop. So there are 3 different notebooks in the workspace, and the one we're going to run this afternoon is this germline variant discovery tutorial. On any featured workspace that has notebooks in it, you'll get this table here of the runtime settings that you should start up with. So when I up here just opened up this and told you all the things to fill out, I was getting it from this table right here. So if you find another workspace you want to run, you can always check for what runtime settings they recommend. And then there's some links to some extra documentation and support if you're wanting to look at this after the workshop itself. All right, so it looks like my notebook runtime has stopped. So if I go back over to my notebook itself, it says it's starting it again already, which is good. So that one stopped. I'm just going to refresh and double check. Yep. So restarting a notebook after it's been paused only takes 2 minutes, which might be another reason why you want to pause instead of delete your runtime after you're done working with it. If you're planning on working again with it soon, it's easier to start up again by just pausing it. But if you're not going to touch the notebook for another like 6 months, it's probably best just to shut it down. So this one's going to take 2 minutes to start up. Make sure that yours are starting up again as well while I go through some other portions of the workspace here.
If it's still running, then we just let it.
Oh.
Sorry.
If it's still running, just go ahead and let it. Good. So another tab in the workspace is the Data tab. And if you click on over to it, you can see these tables that I referenced. Now this particular workspace doesn't have any data in it because we're only running in a notebook, so we aren't running a workflow. We don't really need a table set up. But in a few of the other workspaces, I'll have some tables to show you here. An interesting thing I like to point out is this Files button here. If you click on that, you'll see this is your Google bucket in your workspace. This is where all your files for your workspace are stored. So you can see that there are some notebooks in there, and if I were to click in, I could see these are the 3 notebooks that are listed in my workspace. Then obviously we have the Notebooks tab. You can create a brand new one or use one that we are using today. You've got this Workflows tab. This is where you'll run pipelines from. So when a workspace has a pipeline to run, it'll be there for you. And you'll just click on it and be able to hit launch analysis. And then job history. This is because I haven't run any workflows yet. There's no information, but this is where you'd go to see any currently running jobs in the workspace, any past running jobs to see how it went and what the results were. Again, I'll show you those a bit later. Looks like I'm running again. Is everybody else running as well? Okay. So before it pauses again, I'm just going to have you scroll down to section 1.3, and you'll find this first section of code. And I want you to hit run on it. There are 2 ways you can hit run. One is by clicking this run button here. And the other one is with a keyboard shortcut, which is hitting Shift and Enter at the same time. So that just ran. Everybody successfully run that one? Okay, great. I'm going to go back up to the top so I can talk about the rest of it, just so it doesn't pause on us. All right, so in this tutorial, we're going to go through the variant discovery portion of our pipeline. So what we've provided to you in this workspace is a already pre-processed BAM file, and we're going to, right here, we're going to go through running haplotype caller in a couple different modes to show you the differences. And then we're going to do some joint calling. And then at the end, we're going to explore some variants that we found because they have some interesting things going on there. The very first portion of most notebooks is going to be setup. This can be setting up your files, this can be setting up your environment, any other little things that you couldn't handle as part of the runtime, we do here. Now, since this is the first notebook we've opened as part of this workshop, I'm going to go through all the details of these, but it's going to be the same for every notebook we open, for every tutorial we run. So I'll probably skim through again later. The first section we always put in is the runtime values. So some people don't read the dashboard, or some people don't have an instructor telling them to set the runtime values. This is telling you the very first thing you need to do is change your runtime values. You can open up a runtime with like default settings, and in some cases it'll work, and in some cases the programs may error out. So I think in our case, like GATK would error because you wouldn't have it in the workspace because you didn't enter the startup script. So this one we already did, we're good. The second bit to set up is to check your kernel type. So the kernel is actually this bit right up here. So mine says Python 3, as should everyone else's, but there are other kernel types you can use. If I go to the kernel menu here and select change kernel, you can see there are a bunch of different types you can select. Oh, that's not, there it is. Yeah. So you can run in either Python 2 or 3. We're using Python 3. You can run in R code, or you can run in something called PySpark, which is Python with Spark capabilities, and that's either version 2 or 3. For our purposes today, we're going to be using Python 3. So go ahead and leave that as is, but if ever there's a case where you're creating a new workflow, or not workflow, a new notebook, you'll want to make sure you pick the right kernel for what you're trying to compute on. All right, then this next bit is setting up our files. So we've already run this first command, either using Shift-Enter or clicking the Run button. I prefer Shift-Enter because it's easy to run commands fast. If at any point you fall behind or your notebook crashes and you have to restart, you can just follow along with what I'm saying, and then when the notebook starts up again for you, just hit Shift-Enter through all the commands to get caught up. It's nice because you can get caught up fast and you don't lose the information I'm talking about. So this first command that we ran is setting up some environment settings. So it's importing a library called OS for operating system, and it's setting an environment variable for our workspace bucket. Now, everybody's workspace is unique to them, so everybody has a different workspace bucket. And since we're going to be accessing by copying files out of our notebook, we want to have an easy way to reference that. And by doing it this way, you guys don't all have to copy in the URL to your workspace bucket, which is a really complicated URL because it's randomly generated. And it's like 30 characters long. The next one we're setting is a workshop variable. So we update our workshop materials for just about every single workshop. So what this one is saying is we're in the year 2020 and we're in the month 2, so February of 2020. That's your guys' workshop. So we've run that one. The very next one we're going to do is create directories. These are going to create the places where we output files to. So just go ahead and click on that cell and hit Shift-Enter. You'll notice this one pops up with a little asterisk there. So that asterisk means it's actively running, it's doing something, it's thinking. Another way you can check if your notebook is thinking is up here by the kernel. You see this circle is empty now, and that's because it's finished running. It's no longer an asterisk. Empty circle means it's not doing anything, it's just waiting. Filled circle means it's actively working on something. If ever you see an asterisk here and an empty circle, it means that something disconnected, just refresh your page, it'll fix it, typically. The other interesting thing to point out is that this one put a 1 in the brackets and this one put a 2. So I can scroll all the way down to the very end of this and just randomly run a line. But for reproducibility purposes, we want to know what lines we ran in what order. So that's why it prints out what command you ran in what order. So if I were to go back and say, hit shift enter and run this again, you see it replaces it with 3 because I ran that as the third command. I just did that as a demo because it's not going to break anything, but you don't need to do that. All right, so now that we've created our environment variables and we've set up our folder structure, we're next going to check data permissions. So you're going to do that by clicking on the first command in section 1.4 and running it with shift enter. What this should do is list a bunch of GS URLs. So whenever a file name begins with gs:// that means it's coming from Google storage. So it listed a bunch of URLs there. If it doesn't, it means that you don't have the right permissions. I haven't seen that error in like 2 years, but when it does error, there's like no message later in the tutorial that tells you any indication of why it's failing. So we keep that in there as a check. All right, so if your data permissions are good, you've set up your directories, now it's time to download the data. So go ahead and click shift enter to run section 1.5, that command there. Now, some files and some tools can use files directly from a Google bucket. You don't need to copy it to the local notebook folder, but some other tools like to have them local and they need you to download it. I know we're a lot about talking like just one place and not duplicating storage of anything. But this is a way we can run the tools in the way they need to be run. And then when the notebook's done, we shut it down and all the files are deleted. So anything you didn't need to keep, like these starting files, because you already have them in a bucket, just get deleted so you don't need them again. So this should...
They delete themselves?
When you delete your notebook runtime, everything on it gets deleted. Yep. All right. So everyone should have downloaded the data to the notebook. Have we all done that? Yes? No? Need help? Need help? Yeah, right up here. Oh, great. All you have to do is uncomment that line then and run it in the cell below. That's how you fix it.
Scroll down a little. It's this one, that
Yeah.
bottom line. So you go click in there and get rid of
All right, great. So we've got our data downloaded. The very next thing we're going to do is set up IGV. So as part of your installation instructions, you should have already downloaded IGV to your computer. If you haven't, that link is right there. Please download it now. You will need it for this tutorial. And then you're going to follow these instructions to connect your Google account to IGV. You can click on that and open it, or you can follow along with me on the screen. What I'm going to do is open up IGV. It should look something like this when it opens up. I think it takes a few seconds, so I'll give you a minute. So once you have IGV open, we're going to go to View and then Preferences. So under the View menu, go to Preferences. Once you have that open up, make sure you're in the General section, and you're going to check this button for Enable Google Access. So what that's going to do is allow us to read files out of our Google buckets without downloading them. So that's what we want to do. So you'll just check this box for Enable Google Access, and then hit Save. Once you've done that, you should see a Google menu here. If you don't close the program and reopen it, sometimes it doesn't quite connect. So in that Google menu, you're going to go to Login, and it's going to open up a new browser window for you. You're going to want to select the same account that you logged into Terra with. Basically, what this is going to do is give IGV, tell it that you have the right permissions to access your own Google buckets. If you sign in with a different account, it'll throw a permissions error and say, you can't access that other person's account. So I'm going to sign in with this one and hit Allow. And that should just throw you straight back to IGV. There's no other messages. It just does that. And now your IGV is all connected. So I went to the Google menu here. There's a login option there. That's going to open up a browser window that gives you all your options for logging in. Log in with the same account you logged into Terra with. And then it should throw you right back to IGV.
So if it doesn't connect to the Google account, we just close it and open it again?
Yeah, if you don't see this Google menu, just close IGV and reopen it. All right. So when you're done setting up IGV and you go back to your Chrome window, you'll notice that there's this page open and it just says, OK. That was IGV telling you, yeah, you did it, you logged in. So you can close that.
Very expressive.
Yes, very detailed. So now that we're back in our notebook, we've finished with the setup portion. And it's kind of overwhelming how much setup you have to do at first. But once you have it down, it's pretty simple, and we can get straight into the meat of the tutorial now.
Yeah, tomorrow will be faster.
Tomorrow will definitely be faster. This first day has been a lot of like front-loaded exposition for you. All right. So the very first thing we're going to do as part of this tutorial is run haplotype caller. So we've been talking about haplotype caller and what the command looks like and what it can do. What we're going to do is take this box right here, click in it, and hit Shift Enter to run. Now I'm just hitting run right now because it's going to take, I think it's like a minute to run, and it's easier to do that while I'm talking about the rest of the stuff here. So we are going to be calling haplotype caller on a set of samples today that's called the CEU trio. And specifically, we've taken the samples NA12878 as our mother, NA12877 as the father, and NA12882 as the son. For those of you that don't know, this trio is part of a larger data set where it was, I believe, the mother and father, like the grandparents, and then 11 children.
She's the central.
Yes,
sample,
she's the
and
central
her
sample.
Parents, her husband, and their 8 children.
I thought it was 11.
It's at least 8. Yeah, maybe
Okay.
it's 11.
Big family. And so this NA12878 sample is the most sequenced individual in the world, and she makes a great study for tutorials like this because it's publicly accessible data. And I believe we also use it for benchmarking of tools, and lots of people use it for various other studies just to see how things work. So it's a great example for us to use today.
Yeah, the subline is in Corel, so you can go buy the sample,
Mhm.
run your sequencing experiments, and then evaluate the variants that come out against the data, do the whole pipeline.
Yeah. So just so we keep in mind the identity of our numbered samples, we've just named the files mother.bam, father.bam, son.bam, things like that. So you'll see that, but if you're ever interested, you can look up the CEU trio, and those are the numbers associated with those samples. So this command that we ran right here is calling GATK as our toolkit, and it's calling our tool of haplotype caller. You guys should remember this from the slides, but I like to just point it out again. I believe most tools take in a reference, but it's not required anymore. Yeah. So this first one we're passing in is a reference FASTA file, and that reference is against which the mother's variants are going to be called, and we're going to pass in the mother's BAM with this -I for input. This next one here is a -O for output, and you'll notice with the URLs, the reference and the input are both coming from Google buckets. So we're reading directly from a public Google bucket where this is stored. But the output, we're actually writing back to our notebook itself. So we're keeping the storage in there. And we're just writing it to the name mother HC VCF. This last little line here is not something you need when you're running a full analysis. For our purposes, since we want to make these run fast, we've subset it just to, I think it's 200,000 base pairs on chromosome 20. So we're going to specifically focus our analysis in on there, so these commands will run quickly and we can move through them. So I believe by now everybody should have finished running. You'll notice that under the command itself, it outputs all this text. For any of you familiar with running GATK in the command line, this is your command that you enter in the command line, and this is what's written to your command prompt there. So if after today you decide I never want to use the cloud again, you can still use these commands just as they are. You'll just need to change like the reference to wherever your reference lives, etc. But it's the same syntax and everything's the same. Yep, it doesn't change, no. So this output here is telling you some information about how it's running. And I always like to scroll all the way down to the bottom just to check how long it took. So this particular one took half a minute. So not bad. And the next command here, it's going to use a tool called gsutil, which is a Google storage utility tool, to copy our file from our Jupyter Notebook to our Google bucket for our workspace. So when we run this one, by hitting Shift-Enter, should go pretty quick because there's only one file. There you go. Oh, 2 files. I forgot about the index. And what that's done is it's copied the VCF output of that last command to a place where now our IGV can view it. So we're going to open up the results in IGV. So go back over to your IGV browser here. And the very first thing I want you to do is make sure that your genome up here is set to B37. You might have to click More here, and then you can just type in B37 and load this human B37 one into your reference. So mine is already set. And this is important to do first because it's frustrating, but when you load in your samples and then decide to change your reference, it deletes everything you've loaded. And then you have to reload them. And that's no fun, especially when you've loaded like 20 samples in. So the way we're going to load them in is using this file load from URL. It's going to pop up with this window here and ask you for the file URL. To get that, we're going to go back over to our notebook and scroll down a bit. And there's this command here that's going to print out the file paths that you'll need to copy over to the other thing. So I hit Shift-Enter to run that. And it prints out 2 file paths for me. It's printing out the mother's BAM, and the haplotype caller VCF we just created. One at a time, you'll want to select one line. So we're going to select this BAM here, copy it, go over to IGV, and we're going to paste the URL into there. You can leave this index URL blank. It knows to automatically look in the same location for an index URL, but if your index URL for whatever other reason is outputting somewhere else, then you'll need to specify it. So go ahead and hit OK. It'll take a second to load, and then it says zoom in to see coverage. So basically you can't see anything when you're looking at the whole chromosome right now. Before we zoom in, I'm just going to go back and grab the VCF. We'll copy that over to IGV, do load from URL, paste in the file URL, and you're going to hit OK again. If you're having any trouble with this, raise your hand. Falling behind, raise your hand. Yeah?
It's still loading the version of the genome, so it's just there.
Oh, the genome's taking forever.
Yeah.
That's frustrating. I wonder if it's because of slow internet, maybe? I'm wondering if it's maybe because of the slow internet, because it's
The internet?
pulling the
I don't know.
genome.
I don't know if the machine, because it's short
Maybe.
for me.
Are you on the same internet?
I had the same issues that when I was installing IGV, it didn't load genomes from the first place. I mean, it got an error, so each time that you load a new genome, it has to download it first. So by default, it only has like a bacteria that is a tiny genome,
Okay.
but these ones are maybe larger, so
Okay.
it has to connect it to that to download.
Yeah. All right, then I'd say just follow along with the presentation for now, and when it's done, you can catch up easily. All right. Has everyone been able to load both of these files in? Should look something like this. Good? Great. We're going to go back over to our notebook, and there's another thing I'd like you to copy right above that text we just ran, and it's this bolded section. So those are the coordinates that we're going to zoom into to look at this. So if you copy it and go back over to IGV, there's this search bar in the top for coordinates. You're going to paste it in, and then you'll just hit go, or you can also hit enter. So when you hit enter like this, it'll zoom you into a specific, I think it's like, 328 base pairs window of a region. Up here at the top, we see that haplotype caller called two variants, here and here. If you click on one of them, or actually, sorry, if you hover over one of them, you can see information about that particular call. So at this site, it made a TT call, which is homozygous variant at that site. If you're familiar with IGV, you'll know that this light blue color means it's a homozygous variant. Dark blue means heterozygous, and then blank means nope, it didn't call anything. And it gives you some information about the genotype qualities and some other things about that particular site. Now, if you're moving your mouse around the screen at all, you'll notice something like this. It's really frustrating and difficult. So I like to go up to this little yellow call out, and you just click it and say show details on click. That way, whenever you click on a read, it'll show you the information, but it doesn't like spam you with a million different things. So I just hit show details on click. Seeing a lot of chatter, so I'm going to give you guys a minute. Are we okay? Good? All right, so one question I want to ask you is looking at the calls here and the reads here, you see that this variant on the right, it's got a lot of variants called in the BAMs there. But what about this call on the left? Would you trust it? Haplotype caller making that call on the left. Would you trust it based on the information? No? Why wouldn't you trust it?
Because it's not supported by a lot of reads.
Yep. Yeah, so if if I even go here and like scroll down further in the reads, I think there's like 3 parts that show some sort of variant there. If I click on this variant call here, it's showing it's a homozygous variant insertion of 3 T's at that site. So it's clearly seeing something that our BAMs are just not supporting. Whenever you see indel-related weirdness like this, there's some settings you can turn on in IGV that'll show you a bit more of a complete picture. So I'm going to go up to my view and then preferences again. Go ahead and click on that. And this time I'm going to go over to the alignments menu. And then I think it's like halfway down, scroll. There's this show soft clipped bases option. It's like, I don't know, 2/3 of the way down. We show soft clipped bases. You want to check that and hit save. So when I hit save on that setting, you'll see that this region just lights up with all these mismatches we weren't seeing before. Those regions of the genome were soft clipped, which means by default, IGV wasn't showing them to us. But now with that option turned on, it's showing that there's all these mismatches happening. I guess I should specify, if you're completely not used to IGV, anything that's lit up is a mismatch or a potential variant. When they all line up like this, that's evidence of all the reads supporting a variant. The different colors specify like what the mismatch is. So it's like red, I think is A or something. You can click on one and find out. I don't want to read that right now. Either way, you can click on it and find out more stuff. But basically, lots of lights in a region like that, not good, not a good match. So when our aligner for this particular BAM originally looked at this site, it found that, like for this read here, this much of it matched, but it had some bit here that just was weird. So it often will still match that large portion that did match to the reference, and then just soft clip out the back, the end of the read. Because sometimes something happens when you're sequencing and the end of the read is not as good, so it found the penalty for soft clipping less than the penalty for just not putting it there.
So if you look in the reference sequence at the bottom,
Mm-hmm.
lines up with that variant. Maybe
Yes.
can you zoom in just a little more?
Sure.
Yeah. So there's that long sequence of T's, and then a C, and then more T's. And just like I was describing with the strand bias before, the sequencer reads into the T's, it gets stuck on T, and then the rest of the sequence just ends up being T's, and it gets soft left off because it's probably not a real thing. Some of them look like they might match the reference. You know, there's some bases in there that aren't T's, but they were probably just really low confidence because a lot of the molecules in that cluster were stuck on T.
Mm-hmm.
But that's like the classic homocolumn artifact.
Yeah. All right. Zoom back out a little bit. So these purple ones here, if you haven't clicked on it yet, go ahead and click on it. The purple eyes mean insertion. The wider the insertion, I think it'll start telling you like the number of base pairs in it if you have the most updated version. But if you click on it, you'll see that it shows insertion of 3 bases for that. So based on 3 reads supporting this data, haplotype caller decided to call a variant there. Now, I believe the Broad knows what it's doing, but let's check anyways. So we're going to go back over to our notebook here, and you'll notice that I'm skimming past a whole bunch of screenshots and stuff. It's just things I've been walking you through. Screenshots are great if you're doing this tutorial on your own. So haplotype caller has another feature or another option called BAM out. And what it's going to do is show you the BAM that it created after its realignment, its reassembly step. So we're going to go ahead and hit run on that. So Shift Enter to run. And the reason why we don't run BAM out by default is it makes it run a lot slower. So when you see a region that you really want to delve into more, you want to see what haplotype caller saw, you can add this BAM out, and you'll notice that I also narrowed the region down to just 1,000 base pairs. So instead of that 200,000 base pairs section, we're now just only looking at 1,000 of them. The rest of it is pretty much the same. You got reference, input, the output has a new name, so we don't overwrite the old file, and we're just running that through. When that's done, we're going to go ahead and run this gsudo copy command to copy our output from the notebook back to the bucket so we can look at it. And then we're going to go down here and echo the URL for what you need to copy to open it up. I mentioned earlier there's a reason why we have this bucket thing in there. This is your bucket URL here, this really long, messy string of numbers. It's not the easiest to type. This is why we create variables for it. So we're going to copy this over into IGV. You can go File, Load from URL, and paste it in. I'm actually going to undo some of my things that I had. I'm just shifting tracks around here so that I can see a little bit more on my screen. All right, so now looking at this, we have our top original BAM, and then this is our BAM out that was output by haplotype caller. So looking at this, do you trust the call a lot more now? Yeah? It's got some evidence for an insertion now. And as you pointed out, Laura, since it's a homopolymer T region, it makes sense that it would be a difficult site to call. An interesting thing to point out is also this hard cutoff here for our reads. So we did give it a specifically narrowed portion of the genome, but why did it decide to cut it here and then have a gap and then cut it there? In the haplotype caller talk, we talked about active regions, and haplotype, the tool does this in order to narrow its focus so that it doesn't spend a whole lot of compute time working in regions where there's nothing really going on. So it found this region has something going on in it, which we knew because that's what we were targeting. But there's also another region over here. If I scroll slightly over, you can scroll by just clicking in the track and dragging. It found another region right here with another variant in it. So it did some reassembly at that place. So yeah. Now we see a lot more evidence for variation. And you'll notice that this also doesn't seem to have the soft clips in it anymore. Is that because it cuts them out, or do you know why?
It unsoft clips them.
Unsoft.
Try to use them in the assembly, because if you have a larger event, BWA won't understand a large insertion of soft clipping, but we might be able to reassemble it. So those were just turned into normal mismatch spaces.
Mm-hmm.
Then after that, if they really don't look like they match any of the haplotypes, they might get dropped. Why
Okay.
don't you look at what the depth is on the BWA BAM versus the BAM out?
This one? How would I check that?
If you click on the coverage track.
Ah.
Where's your coverage track? All right, wait.
It's like hidden. Ah, there it is. 30? Yes.
I'm so sorry, can you repeat what BAM out means?
What it means?
What make BAM out in the Jupyter notebook?
Yeah, so back in the notebook, if I go back to the command up here, you have this dash BAM out option that we added. So that's creating the BAM output for that particular run. It's showing us what haplotype color reassembled the region to be. All right, so it looks like 30 versus 25, so it might have dropped something. Read depth. Okay.
Question.
Yeah?
What does the pointy end mean?
So each of these gray bars is a read, and the pointy end shows you what direction it's running. So this particular read ran from this direction, this direction. This read here ran from here to there. So that's what that means. All right, so there's more to this than meets the eye. If you look at these purple eyes here, you'll notice that they're kind of different widths. So I'm going to go ahead and click on one here, this top one. Oh, it's stuck. So this top one saw an insertion of 3 bases, but this little skinny one underneath it only saw an insertion of 2 bases. So there's some interesting ways you can work with IGV to get it to tell you a little bit more about what's going on. So what we're going to do is, in our track here for the debug BAM, we're going to right-click, and it's going to pop up this big menu of things we can do with the track. The one we're interested in is this color alignments by, and then we're going to choose read group, which is right there. Go ahead and do that. So color alignments by, and then read group. What this does is it changes the color into blue and red. Let's click on a red one and find out more about it. Oh gosh. All right, so this is one of the red reads, and its read group is listed as artificial haplotype read group. As part of haplotype caller's assembly step, it creates all these possible artificial haplotypes at that site so that it can decide what the proportion of matches to that haplotype is versus others. And these artificial haplotypes are actually put into the BAM out so that it can measure how much support there is for this haplotype versus this haplotype versus this haplotype. So if I click on a blue one, you can see the read group listed is H064.2, and that's something off of the library sequencing part. So the artificial haplotype has an artificial read group because it wasn't created by a sequencing machine, but this one has a real read group because it came from a sequencing machine, and it's a real read. So these red ones here, they're not real reads. They were artificially created by haplotype caller in order to track how many reads support this haplotype, how many reads support this haplotype, how many reads support this haplotype. So it's using those as a way to store the information so that it can, in the end, make a call. And when you select a BAM out, it outputs these artificial reads as part of it.
Are we going to
Color them?
Yeah.
Yeah, we're going to color them in different ways.
Okay.
Yeah, no worries.
So in that case, all the reads that support that are in one direction?
I think what we're getting to next will help you with that. So we're going to try something a little different. Right-click on this track again, and I want you to sort alignments by, and then choose base. Sort alignments by base. And what that does is you'll notice that all of these are the same width. They're all your 3 insertion. This is all your 2 insertion, and then some other stuff down there. So it's nice to do that. And then if you now color your alignments by, and we're going to choose tag. So you're going to go color alignments by. You're going to select tag here. And it's going to pop up this menu. You're going to enter the tag HC, and that stands for haplotype caller. And what it's going to do is color your reads based on which artificial haplotype they support. So I hit OK. I'm just going to squish this a bit. Does everyone have their IGV looking like this? Need help?
If we switch option, color alignments by,
Color, alignments by, and you're going to choose tag. So color, alignments by, tag, and then when it pops up, you'll type in HC, capital, all capitals. Color, alignments by, then tag, then this is what you enter in the pop-up. Are we good? Haplotype color. Yep. What I just did? For what I just did, where the tagging? Okay. You're going to right-click on this track. So I right-click, pops up this menu. You're going to see the color alignments by option. You're going to go over and down to tag. When you click that, it pops up a little pop-up, and you just type in HC. All right. So now what this is showing you is it's highlighted the reads in 4 different colors. So you've got red. You know your artificial haplotypes are at the top because we just clicked on those and saw it. You know this top one is red, so we're saying anything that's in the real reads section that's red shows unequivocal support for this haplotype alone. Anything that's highlighted as yellow, or maybe that's green. Yeah, it's green, it's just washed out. Anything that's highlighted in green shows unequivocal support for this green artificial, this 2 base pair insertion artificial haplotype. And then this last one here in purple is our reference call. So anything highlighted in purple shows unequivocal support for the reference. If you scroll down, you'll see there are 1, 2, 3, 4, 5, 6 red reads, 2 green reads, and no purple reads. But there's a lot of gray reads. When they're gray, it means they could support either. You remember this is a homopolymer T region. The ones here in gray, they could support a 2 base pair or a 3 base pair insertion at that area. It could go either way. That's why it couldn't decide which one it was, just because it's such a short read. But based on this information, haplotype caller saw 6 pieces of unequivocal support for this 3 base pair T insertion. Given that information now, do you now trust the homopolymer T or the 3T insertion call it made? Make more sense? All right. Any other questions before I move on to the next bit? All right. So we are maybe a third of the way through this tutorial right now. It's nearly 4 o'clock, so if you have like childcare pickups you need to do and you need to go, feel free. I'm going to keep going through this. You can absolutely continue it on your own at home, but I'm going to continue to teach it for those of you who are willing to stay a bit later. We're running a little bit behind from some other stuff, so we're going to move right along. Going back over to our notebook here, I'm going to go back to where we were. Okay, so we just ran this. I just walked you through that part. So now we're on to the GVCF workflow. Up until now, we've been running haplotype caller in normal mode, and we've talked your ear off about the GVCF mode, so we're going to run that now. This first command here runs haplotype caller, and we've added one additional option, which is -ERC GVCF. That stands for Enable Reference Confidence Mode, and then we're picking the mode GVCF. If you'll recall from John's slides, I believe, there's a base pair resolution mode, but typically GVCF is what you want to run in. The other change we made is now the output is no longer a just VCF, it's a .g.vcf. And by having that syntax at the end, the .g.vcf, it tells later tools that this is a GVCF and not a VCF. So although a GVCF is technically a VCF, it has some additional information which it can take advantage of if it knows it's a GVCF. So we're just going to hit Shift-Enter to run it. Shouldn't take too long to run. We're again running this on the same mother's sample. We're just going to see her all throughout this process. And when that's done, you'll just copy it out to your bucket. An interesting thing is you can actually run multiple commands at the same time. It'll just queue this next one up to run whenever that one's done. So if you have a set of like 5 commands, you can go enter, 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 enter.
What does ERC GVCF stands for?
Stands for Enable Reference Confidence Mode, and then GVCF is the type of mode you're doing. So, yep. All right, so that looks like it copied for me. And then next we will print out our paths. So in the interest of time, I just made you run that once for the mother, but now we're going to do some joint calling. So we've provided both the father and the son's GVCF. You could absolutely generate them yourself, but just this is easier. So once you've printed out these file paths, I'm just going to copy the first one while I'm there. But before I start loading in new things, you might notice that our screen's pretty crowded. So what we're going to do is start a new session. So go to File, and then say New Session. All that does is clears all the tracks you currently have loaded, but it leaves the genome you have up just fine. So hit New Session. I'll put that down at the bottom. And we're going to go File, Load from URL, and you're going to copy in all 3 of the GVCFs, one at a time. This is probably the most tedious part of it. I know IGV is working on a way to just copy in multiple URLs at a time, but they don't have it there yet. Go back, copy this. For those of you who have this loaded up, and keep working if you're not, I just wanted to talk a little bit about what you're seeing here. So these gray blocks here are your non-ref blocks. So when we were talking about it in the presentation, we mentioned how it groups together sections of the genome that we didn't see any evidence for variation, and we grouped them together by similar genotype quality values. So for a section like this, it apparently varied either back and forth or varied wildly between genotype qualities. But for like this section here, they all had a very similar quality, so we were able to group that section together and save ourselves some file space. The other track you see here, which is this one, and this one, has variant calls in it. So if you recall, I said the dark blue is a heterozygous call, and the light blue is a homozygous variant call. So actually, let me just double check I'm in the right coordinates. Because I've been zooming all over the place. There we go. That looks a little bit better. stretches it out a bit more. So given what we know about these samples, is there anything suspicious to you guys right away about those variant calls?
The father doesn't have variation.
Yeah. Yeah, so we're seeing a son with homozygous variant sites, and the father has no variants called at either site. So either this isn't the dad or something's wrong.
The father.
So what we're going to do now is go back over to our notebook. And this is just another way you can look at a GVCF here. It's going to print out the top 100 lines of the file. I find it kind of interesting to show this because you can see these are the GVCF blocks. So I mentioned how it groups them by genotype quality. So the bins that we did here are from GQ2 to GQ3, so they're pretty small. But if you want to change them, you can change it. I think this is the default though, right?
It is, I'm
Yeah.
gonna change it.
Yeah?
Yeah, we just gotta get the rest of our pipeline
Ah.
updated.
There you go.
Yeah, it's like you get really high resolution in the low quality data versus just files.
So if you scroll all the way down to the bottom, you'll start seeing some entries for your reads, or not reads, I should say. You've got your ref blocks and you've got your variant calls. So this first one here is a ref block, and it starts at position 10 million, and it ends over here at 10 million and 8, so it's only 8 base pairs long. And it gives you information about the genotype, the depth, the GQ, all these other variants, and it shows you all that information there. Now, for this particular site, there's a ref allele of a T, but the alt is listed as non-ref, but there's no specific alternate allele. So non-ref just captures all possible evidence we have that it could be anything else. In some cases, our joint discovery or our later steps can help recover sites that maybe we didn't have great data on, but maybe we have a lot more data elsewhere. No, it can't do that? What does it do?
The goal is to be able to assign a likelihood to alleles that you haven't seen. Because you will discover alleles in your other samples that were not in all of your samples, and to have that squared off matrix from John's joint genotyping presentation where you have probabilities for every variant for every sample, you need to fill in the blank with something. And so we get this reference confidence by looking at the mismatches and the soft flips and the base qualities at that position, and we add them all up and we say, what's the probability that there was something here, but by a series of errors it looked like the reference. And probably is usually pretty low. But and that way we have a number to assign to any alleles that we discover in the rest of the cohort.
There you go. I don't see any variant sites here. Yeah, a variant site would have a lot more information. I wonder if I could just expand this.
Do a rep on, I don't know if the person understands it, forget it. Just rep the whole file, and then do 0 \//1.
That?
And then this
Other way.
And that should pull out the heads.
Oh, there you go. Yep. So this is what a variant site looks like. You'll see that it has a start position, but there's no end position listed there. And it also has a lot more text there. So if you're just scrolling through the file really fast, you can pick these out pretty easily. It's got all the evidence captured for this alt allele T for that particular case. So the next step we're going to do is consolidate our GVCFs using Genomics DB import. So what this is going to do is turn our 3 GVCFs into a Genomics DB data store. And the first step is to remove all previous references to that. The reasoning being it has to be a unique name, and if you run this notebook before and then try to run it again, it'll error and say, this already exists, we won't overwrite it. So, there is? Oh, good to know. So you'll run that step, and then you'll run this GATK genomics DB import. You see we're passing in 3-V files, which are our mother, our father, and our son's GVCFs. And then we're outputting it to this genomics DB workspace path across, again, the same narrowed region of the interval we've been working with for most of this. So when that returns, we're going to create a soft link to the sandbox. And the first line is to remove the soft link in case you've run this before. And then you'll run a select variance. This particular step is not part of the pipeline. What we're doing here is taking that genomics DB data store, we're opening it up, and we're going to see, hey, are my 3 samples in there now? Did I really do what I said I did? So when that runs, I'm going to run this cat command, which is just going to print out the top 100 lines. And if I scroll all the way down to the very bottom here, you can see that this file has 3 samples in it. At the very end here, after format, it lists the sample IDs. So we have 3 sample IDs there, NA12877, which is our father, 878, which is the mother, and 882, which is the son. So yes, the genomics DB data store has combined them all into one thing.
And notice now that the genotypes are no calls.
Yes, that is important.
At this point, we haven't done the final genotyping. We don't know which alleles are the high quality alleles that we want to keep. And so we just nullify the genotypes so you don't look at them and say, oh, this is ready to go. I can start analyzing this. It's just to make sure that you know that there's an additional step here.
All right. Then lastly, to do that joint genotyping, we're going to run this genotype GVCFs command. We're passing in only a single file because remember this tool can only take in a single file. So it works if you're running on one sample or you have to combine it into a GenDB data store like that. So we'll hit Shift Enter to run it. And what this is doing is now doing the joint calling. Yes?
Why is there a right error?
Oh, the red text on this?
No.
That always happens.
In this cat right error? Yeah. It's saying that we found the end of your command and it's not happy about it, I guess.
Oh, okay.
Yeah, it has to do with like when the notebook calls out to the shell on the VM and it's running a command there. When it's done, it's still looking for more data.
Okay.
Yeah.
It just always happens. It's not a big deal.
Yep. Good question though. All right, so when that's done, we're going to copy it out into our sandbox. And later we're going to open it up in IGV. So this is one command we're not running today just because we want to keep this running quickly. But when you make the call of running haplotype caller in normal mode versus running it with GVCFs in multi-sample mode, the calls you get out should be equivalent. There's some nice new qual score models that you can use with genotype GVCFs.
That should be default.
Default now? Okay, great. And then you can always output a BAM out to look at it. But that particular command you can run on your own time if you're interested in it. Then we'll open this up in IGV to take a look. So copy that URL, and we're going to go over File. And I'm just going to start a new session because these GVCFs take up a lot of space. We're going to go File, Load from URL, and then paste it in. So at this particular site, we see the final genotype calls do match what we estimated in the GVCF. You see that the father is blank and blank, the mother is het and homvar, and the son is homvar and homvar at those sites. Now, again, doesn't make sense inheritance-wise, so there's a little bit more we want to dig into here. So if I click on this blank section where the father's first variant is, we can find out a little bit more information about what's going on at that site. For him, he called a genotype of GG. It's a homozygous reference site, so it seemed like they were pretty, they saw no evidence for even a het. But then if I go down here, you can see the genotype quality is 42. So that's the initial prediction based on the information it had at the time. The other one to pull up is this white bar here. And this one is interesting because instead of calling a reference here, it actually called a no call, which means it didn't see data for a reference, it didn't see data for a variant, but something's going on at this site. Something could be going on at this site, it doesn't know. So it has a no call there.
There is something going on at that site, it just can't identify
Yes.
what it is, which is why the likelihoods are even for homozygous reference or het or homozygous variant.
Yes.
It usually happens when the region can't be assembled.
Mm.
And then as we go through and we look at each position to evaluate the reference confidence, then in the pileup, clearly there's something going on, but we don't allow the reference blocks to be hets, because that doesn't make any sense. So we just say, oh, there's something here, and I don't know what it is. I don't have any confidence.
Mhm. Yeah. All right, so going back over to our notebook, I wanted to show you this table here in the notebook itself. So this table is talking about that leftmost variant here, where the father is a homref call, but the mother and son have things that don't make sense inheritance-wise. Based on all these different colors, we saw a variety of inconsistent or failing filters calls with a variety of different mappers. Yeah, these are all mappers. So this is an ambiguous site. This might be something that needs further digging into if you really wanted to find out more about the father. The one that we're going to primarily examine for the rest of the notebook itself is the rightmost site, because I think we can do something about that no call. Scrolling on down, this is a little bit of a preview for tomorrow. We're going to start talking about genotype refinement. So genotype refinement is going to take probabilities of the population and probabilities from the pedigree information, because this is a related trio. And it's going to apply that, and we're going to see if that changes our calls. So we're going to go ahead, and I'm just going to do Shift-Enter to run all 4 of these commands in a row. It's going to start running, and then it'll run them one after another when it finishes the first one. All right, so I have my 2 outputs. So this first file, trio CGP, encompasses the posterior genotype probabilities after we've applied information from the pedigree. So we're telling the tool that these samples are related. Does something change? So let's load from URL, and we're going to paste that in. And looking at it, it didn't change the father's calls. So now we're going to go back here, and we'll grab the second one we ran. So this trio CGP underscore nomad, in addition to the pedigree data, also added in some population information. So load from URL, paste that in. And you see that with the added population information, it does actually change the father's call at this rightmost site. It doesn't change anything for the left site. Again, it's a difficult site to cover. But we did see some changes with this one. So if I click on it, actually, I'm also going to click on this one to see what changed across all of it. So this first one was our before. It has a no call. Second one, also no call. It adds something at the end. There you go. So its PLs are all 0, which means it couldn't decide between any of the genotypes. But then at the end, it ended up calling a homozygous variant there with a quality of 72, meaning that given the population data and the frequency of this particular allele in the population, and the fact that there was difficulty calling at the site and the pedigree information, it ended up changing the call to a TT. So that third column in our overall GATK pipeline, where we're doing our filtering and our refinement, is pretty important to rescue sites like this. All right, let's head on back to our notebook. I think we're just wrapping up here. All right, so the next step is we're going to compare the changes between our original call set and our after refinement call set. We want to compare these metrics to make sure that our refinement didn't mess up the calls through the call set. So this first command here, collect variant calling metrics, is going to take in our initial raw VCF as well as a DB-SNP resource, which is a resource containing a whole bunch of variant calls. And then it's going to output our metrics to our notebook. And those metrics can be printed out using this line. So you can see it lists it by your sample alias. So this is why I mentioned the numbers, because they kind of do come up. But you've got your son, your mother, and your father there. You've got your total SNPs listed, your TI-TV ratio, your indels listed, your insertion-deletion ratio. These are all metrics we're going to discuss a lot more tomorrow. But I wanted to show you how they change for this particular case and give you a little bit of an overview on what they mean. So this second section here that I'm running of collect variant calling metrics is running on our Trio CGP. So this is after the pedigree information has been added. And it outputs our table here. So I'm just going to double click on that to collapse the output so that I can see them both on the screen. So if I look at the metrics up here, this was before refinement and this is after refinement. You compare the total number of SNPs between the two. You see that 3 SNPs were rescued for the mother after refinement and 4 SNPs were rescued for the father. Looking at total indels, which is the 66, 69, 64, we see improvement across the board, rescued more indels. The other two we have here is the DB-SNP TI-TV ratio. So I believe, yes, so for whole genome samples, we expect the ratio to be between 2 and 2.1. The initial one here, we don't see it between that. The after one here, we don't see it between that. But we do see some improvement towards that with the father, especially. Oh yeah, and the mother, the father and mother. Yes, so the reason why it's kind of a little bit difficult to compare is because although this is a whole genome sequence sample, it's got that coverage profile, we've only subset it to 200,000 base pairs. So we don't really have the whole genome. So that's why it's a little bit wonky, but we do see improvement after this genotype refinement. The last one is the insertion-deletion ratio, which we expect it to be about 1 for common variants, and then it's closer, I think it's like 0.2 to 0.5 for rare variant studies. So if you're searching for a rare variant, you tend to see that ratio. For this one, we see it at about 1. This one brings it down a little bit closer to 1, up a little, up a little. So it's kind of hovering around the same area. It's not like great confirmation, like this made it so much better, but it also isn't, it hasn't turned that to like 3, which would tell us, hey, we did something that we probably shouldn't have done. So this collect variant calling metrics portion and genotype refinement is really great for refining your data set and making sure that the refinements you make are good to make to the call set as a whole. So we're going to talk about that a bit more in depth tomorrow, but I think it's interesting to take a look at those while we have the data in front of us. This is the end of our notebook. How do we feel about notebooks? Yeah? It made the commands running easy, I hope. We used to have to make you type everything, so this is easier. Yeah. So if you have any questions about this or about anything that happened today, feel free to ask them now or come up to us afterwards. But otherwise, we'll see you tomorrow at 9. We're starting.
Well, just one more thought.
Yeah, more thought.
There's still unfiltered calls.
Mhm.
Using the family information and the population priors helps you improve the calls a little bit, but for example, having a lot of heterozygous variants is a very common error mode, which we wouldn't be able to fix from the pedigree information because that's a perfectly valid combination of gene types in the family. And from just 3 samples, we don't know if they're in party-Weinberg equilibrium or not. So to really do the best job filtering these calls, you want to call them in the context of a larger cohort and also run some filtering.
Yes, yeah, absolutely. Yeah, we'll be discussing filtering a lot more tomorrow. It's too big a topic to tack on to the end of this, I'd say. Oh, and if you have a Windows machine and you couldn't get Docker working, don't forget that document I gave you about installing and testing GATK. So, all right. Thank you, everyone. Oh, yeah. If you'd like, you can pause your notebook by just hitting this pause button. Don't delete it. Just pause it.
Okay.
Yeah, we're going to be using the same settings tomorrow, and we don't want to spend another 15 minutes starting up your notebook. Yes.
So when using the DVSNP, DCF, that
Yes.
input data file, is it, you can actually obtain it from DVSNP per se? Like the, they're like those VCFs, there's
I
like,
believe so.
so you can actually use like SNP info from, is it like the,
So DBSNP isn't the greatest truth set. We talk about it a little bit more in the call set evaluation talk tomorrow.
no.
Because it's a collection of all variants that have ever been reported, and there's not really any,
Yeah.
they don't check
Yeah.
how good it was.
It's like,
I believe we got that VCF from where you mentioned, but if not, if it's not there, if it's not easy to access, you can always ask us on the forum, like how to get it.
okay.
So I'm sure we have some resource somewhere about where to get it.
Because I know that there are certain SNP data, like VCFs, that, perhaps are more updated. Like,
Yeah.
Yeah, more recent.
Yeah.
That,
Yeah, that's a good point.
especially if you're doing, or trying to get information for disease identification or doing something like that, especially filtering.
Yeah.
If you're kind of like fishing for something and you're using something that's kind of outdated.
Yeah, you
But I
want
was
something.
wondering if you could actually use something.
Yeah,
that
you
is not-
can use any type, not any type. You can use a lot of different kinds of truth sets in that case. Sorry, I'm just looking for the line where I use it because I want to write this down. What are some of the other resources you mentioned?
No, I didn't mention that.
Okay.
No, but my question was actually that it was going in that line.
Yeah.
Like using any other like disease-associated VCF.
Okay.
Yeah. Because I know that some of them, it's been kind of a while that I've done that, but I know that there are others that you can actually